Hi, everyone. I'm Gary Nell, the chairman of National Geographic Partners. For 132 years, National Geographic has been about science, exploration, and storytelling. And I can't think of a better occasion than to celebrate the 50th anniversary of Tektite 2. I want to congratulate Her Deepness, Sylvia Earle, of course, our hero, and all of the other members of that historic journey who broke barriers to give young women and girls an opportunity to realize their own potential in science, exploration, and storytelling. National Geographic was with you then and will be behind the future generations of young women and girls to reach their potential in the future. Thanks so much and congratulations.
Well, hello everyone. Thank you, Gary, for that resounding message. Definitely near and dear to our hearts. And welcome back to Tektite 2020, Women of Sea and Space, celebrating the 50th anniversary of the all-female Tektite 2 Mission 6. My name is Dr. Tierney Thys, and I'm co-hosting this event with Dr. Sarah Jane Pell. I'm coming to you from Central California. And I'd like to take just a moment to recognize and acknowledge the native peoples of this area, the Rumsen, Esalen, Ohlone tribes that lived here before me. We have had an absolutely phenomenal opening session with this conference that has transported us back in time, down into the sea, far out into outer space, and deep into the lives and missions of aquanauts and astronauts, researchers, and artists. It's been quite a wild ride, and we're just getting started. Now, during the opening session, we've reflected on our Sputnik moments, on our, how the Cold War sparked undersea habitation experiments. We've gotten to know the original aquanauts. And we've reflected on how we are all citizens of the reef and crew members on Spaceship Earth. Now, at the heart of all these endeavors lies our ability to tell stories. Stories are our way of communicating our place with each other, our place on the planet, and for allowing us to share our connections to each other in both place and time. Today's panelists have wildly creative ways, different ways of telling their stories, and I'm so excited to welcome them all. They're coming from all different parts of the globe. We have Alina Szmant, who will be speaking to us from the East Coast. We have Kakani Katiji, who's just up the coast here in California with me. And we have Rachel Butler Scott, who is all the way over in the UK. So this is the wonder of today's bring everyone all together. We can lower our carbon footprint because we're not flying all to the same venue, but we can all be together here and with you. And we are so grateful for you tuning in to join us with this session called Stories from the Sea. Now, I'd like to remind you we are streaming live on Facebook and YouTube, and we welcome your questions. We see them coming in, and time allowing, we will ask them of our wonderful panelists. So let me start with introducing Alina Szmant. If you were tuned in this morning, you got to meet Alina. She's one of the original female aquanauts of the Mission 6, Tektite 2 Mission 6, and has quite a number of accolades to her name. She was a pioneer in the study of physiological ecology and reproductive biology of Caribbean reef corals. Spent a lot of time on those Caribbean reefs. And is now a retired professor at the UNC, University of North Carolina in Wilmington. During her last 10 years at UNCW, she led the development of an innovative diver portable underwater respirometer. And she'll be talking to us a bit more about that today, go into detail that she didn't have time to do in the morning panel. Alina has served on the Scientific Technical Advisory Committee for the Water Quality Plan for the Florida Keys, was awarded the AAUS Scientific Lifetime Achievement Award as well. So many accolades. So Alina, I'm so excited to hear you and have you join the panel, and I'll just hand it over to you.
Tierney, I want to thank you so much for including me in this commemoration of Tektite 2. It's really been a pleasure and an honor to be part of it. So I prepared some slides to sort of talk about that, and if you're ready, I can share my screen. Let's see the technology here to give me a second to do all of this.
Yes, it was
okay.
such a joy listening to all the Aquanauts speak this morning.
Okay, so I'll start by saying it. I can't believe it's been 50 years already. Like, where did my life go? Well, I've done a lot of things throughout my life, and I guess this has been an opportunity to reflect, how I got here and what I did along the way. So I spent a lot of time in the Caribbean as a child and in my formative years, because my mother was Cuban. We lived in Cuba for a few years, left there, went to Puerto Rico. So I was always around the ocean, but not really, marine biology was not a thing back then. And during my high school years, I had a chance to do a field trip to a marine lab, and that was sort of interesting. And then the summer of after my first year undergraduate school at the university, I took a summer course in marine biology at a very small primitive marine lab on the south coast of Puerto Rico. First time I'd been scuba diving, the professor Dr. Peter Glenn strapped a tank on my back, took me diving. I was freaking out. My ears hurt. And then a parrotfish swam by, and it was like, that was it. I was hooked. And I knew that's what I wanted to do for my career. So I changed my focus in my undergraduate studies towards more biology. And when I finished, I went to Scripps Institution of Oceanography, where then I got a good intensive dive program with Dr. Jimmy Stewart, well known for his, the Scripps program was a model dive program at the time. But I was very lab-oriented in my research focus because my father was a chemist and I was going to be a chemist and all that kind of stuff, little girl doing her daddy's footsteps. The summer of 1967, I got to participate in an NSF-funded training program in Hawaii. And with all the coral reefs around us, I ended up working on Hydra. It's a little tiny green freshwater animal living in glass dishes. So I did do some diving, but that wasn't, again, the coral reefs, while everybody knew that I knew something about corals, I wasn't really tuned into the underwater world. And then along came Tektite. So the spring of 1970, my fellow graduate student, Ann Hurley Hartline, and her husband, Peter Hartline, were alerted by one of the faculty that there was this program that was looking for 2 women to join, complete a team of underwater scientists. So they had a project in mind, and they recruited me because of my expertise in the Caribbean to help develop the project. We submitted a proposal, and lo and behold, we were selected. And then started all the publicity stuff. You can see here this, I always laugh when I see this bottom right-hand photo. You can see we're all wet. This is a photo op by the publicity team. They made us wet our wetsuits, our hair dry. There's no tank on our back. So that was sort of notice of things to come. For a young person, I was 23. This was, a really whirlwind experience. All the whining, the dining, the photo ops, the celebrities wanting to meet us. We were followed by cameras everywhere. And by the way, I want to have a shout out here to 2 principals of the Tektite program, Jim Miller, who was the director of the Tektite program and fought for the women's team, and Mark Silverman, who was the public relations person for General Electric. And they were 2 men that really supported the whole idea of women being part of this whole program. So I'm not going to talk too much about the mission itself or the research we did. Time flies fast, and I do want to try to get to Kiss Me, our new instrument. So what did I get out of Tektite? Well, all those hours and hours and hours underwater. A lot of it was done working. We worked really hard on our project, but a lot of it was looking around and asking questions about everything we saw. So my focus for my research sort of shifted from being laboratory physiology and biochemistry to trying to ask questions that were field-based. And I just want to go over just a couple of the more salient ones. As a soft money researcher at the University of Miami, I was very proactive in any RFP, no matter what it was. I worked on seagrasses, I worked on fishes, I worked on coral-eating snails, all kind of different things, nutrient physiology. But one of the major areas that I became known from that required huge amounts of underwater time was elucidating the secret sex lives of corals. And actually, my first NSF proposal, I put in sexual reproduction of corals, of Caribbean reef corals, and there was like, no, that's, don't use the word sex when you're trying to go for research funding. So they suggest I change it to just reproductive ecology of Caribbean reef corals. And when I started doing this work back in the late 70s, early 80s, nothing was known about this field because it took a huge amount of effort underwater, making observations and also bringing samples to the lab and analyzing. So I always combined both methods, field and lab. Finally, once we narrowed down the window when these corals were reproductive, we developed nets and things like that so we could try to capture the gametes. It took several years to narrow down the timing. All of the spawning for the Caribbean happens, for any region of the Caribbean, happens in 30 to 40 minutes at some odd time of night, predicted by the lunar phase. So it took a lot of trial and error before we narrowed down. The first time we actually observed it, then we could start, working backwards and forwards to try to narrow down the timing. And over time, we learned how to culture the larvae and settle them, and we've used it for coral restoration. So this has been the basis for a whole bunch of scientists. There's a lot of young people that have taken up this banner that have the energy to spend the long nights and days doing this kind of research. So I did it for 25 years, and then now I can read what other people are doing with it. And another area that leads me to what I've done for the last 10 years was interested in studying the physiology of coral calcification. Coral skeletons are very important for climate change research and paleoceanography because of the density bands they have in their skeletons. And we didn't know much about why and how they make it. What are the factors that initiate that? So we were trying to do a kind of physiology to try to understand why they make these density bands. A lot of the early studies were, I feel terrible for the poor corals. We went go out and took cores and brought little pieces back to the lab and tried to do experiments there, but we knew there was a lot of artifacts, and we took the research vessel out to the reef and tried to do it on right there above the reef, but still we had to puncture holes in the corals, and of course that's dead coral after that. We used an underwater respirometer developed by Jim Porter from University of Georgia, but again, we had to take big pieces of coral from their natural original donor colony, and with corals going downhill, that just became a non-viable approach anymore. When an animal is suffering, you just don't go out and start digging chunks out of it. So in 2009, I was fortunate to have an opportunity to apply for funds from NOAA's Cooperative Institute of Ocean Exploration Research and Technology, and they funded me and my collaborator Rob Whitehead to develop a whole new technology to assess coral health in a non-destructive way. And we developed a new instrument which we named KISME for coral in situ metabolism. Its intent is to be non-destructive. It's sort of like when you walk into the doctor's office, they do an EKG or a blood pressure cuff, they don't start cutting you up. And the instrument is potentially very valuable for physiological and ecological research, ocean acidification studies, environmental monitoring, conservation, etc. It's still new. There's only a few people working with it so far, but we are commercializing it so that it can become widely available. So I'm going to show this little short video that tells you a little bit more about KISME. I want to interject that there was a huge amount of technology that went into developing this electronics, and I'm a garden variety marine biologist. So we benefited from collaborating from a number of engineers to help us with this. And it took us many years to get it right. Whoops. Okay. So anyway, I screwed up there, so I'm just going to go to try to drag this along. Okay. So the other part that we're really proud of is trying to figure out how to attach this to the coral. I thought this was going to be the most difficult part, and it turned out that once your little internal pump is working, it just attaches it to the coral, and even though we had a lot of turbulence, it stayed firm.
Alina, I'm just going to pop in as you share the video. The sound isn't coming through for us, so could you narrate a little bit as you finish the video?
Okay, I'm sorry. I thought you guys were hearing the sound. Okay, let's see. I can very quickly go back to here. So this is what we call the head. It's attached to the coral, and it has neoprene seals that isolate a small volume of water. And the retractors around the edges, can you see the pointer, the mouse pointer, helps hold it down against the coral. And we use it for algae, for settlement plates. One of the innovative features is that we operate it with an underwater tablet. I think this is the only instrument that I know of that uses Wi-Fi to communicate with an instrument underwater to control it. The electronics took decades to develop, or not decades, but many years. A lot of technology there, and I'm not an electronics person. Let me see. Let me keep going here. I'm sorry about the sound not. So one important feature is we have a sample loop inserted in the circulation pattern that we can withdraw samples for doing alkalinity, for measuring calcification, for introducing toxin, or acidified seawater. The tablet produces, we have proprietary software that took a lot of effort to develop that produces a graph, so we end up giving a live version. We can see exactly what's happening in the incubation. Let's see, and here's some video just showing how it's used. So it's all mounted in a basket so that it's easy to swim around with. These are some NASA, actually, people. I was part of the NASA team, NEMO team, swimming around. Here's Rob attaching it to the coral. The idea is that we can move around with it and go from coral to coral as we want to collect data. And at the end, you just pack it all up and send it to the surface and move on to the next coral. So it's meant to be totally diver portable. So, okay, so over the years, I did many different types of research projects, but one of the things that I started to focus on in the last 20 years was sharing my knowledge and enthusiasm for working underwater with young people. At the University of North Carolina, Wilmington, we're very engaged with student projects, and I developed these 2-week, 3-week, 2-month courses, taking students into the field to learn how to work underwater and how to do research, how to ask valuable research questions, and how to plan them out and execute them. So that was really rewarding. And I still am in communication with, I've had maybe over 100 students that I've trained how to do science underwater. So not all of them have gone on to graduate degrees and on, but many of them have and are being valuable scientists. So this is my last slide. Depending on, let me see, I got to end sharing here. Let me see how I do that. I have to go back here.
Thank you so much, Alina. It's wonderful to have the context that you bring to this whole study, and also just the laying the groundwork for being able to understand where we are today with coral research. I mean, these tools that you've helped develop, and the insight and the observations that led you to those tools. Would you have ever believed that we would be at the state that we are with coral reefs now?
You know, one of the challenges of developing this instrument, because, you know, again, we started from scratch. We started with pen, paper and pencil. Like, if we wanted to do this, I honestly didn't think we were going to be able to do it. It wasn't my first choice of a project to do, but okay, once I was given it, okay, we'll do our best. But the technology kept evolving, and so new tools like 3D printing. Who knew about 3D printing 10 years ago? And so, like, halfway through our project, the engineer up in Boston said, hey, there's this new method. You know, instead of doing machined aluminum, we can do 3D printing. So therefore, we can have all these channels that are skinnier, that are faster. We can have smaller pumps. So, you know, it was a real process. It's nothing I ever thought I would be involved with, but it's been fantastic. And my goal is now is that this instrument be useful for the conservation of coral reefs, because coral reefs are in trouble.
Yes, no, absolutely. And having the more non-invasive tools, the better.
Yeah.
Well, thank you so much, Alina. Please.
Do you have time to show the little comedy sketch, do you think?
Well, not right now. We'll hopefully get to it if we have time at the end of the session. But I'd like you to speak about that when we open up our to have all three speakers together to talk about ways in which we can tell stories. So that'll be something for the panel. And so I invite you to stay with us. And at the end of the panel, we'll all come together and we'll have a discussion. Thank you so much. So our next speaker is Dr. Kakani Katiji. She was originally an aerospace engineer and was interested in reaching into outer space, but turned her gaze towards inner space. Kakani received her PhD in bioengineering at the California Institute of Technology, and is now a bioengineer at the Monterey Bay Aquarium Research Institute in Moss Landing. She's a principal engineer there. Her team is doing amazing things. They're developing state-of-the-art technology for platforms that include all sorts of advanced imaging tools to measure biological physical interactions, electronic tagging packages. And then I do want to mention that Kakani has multiple talents. She's also an US international figure skater. So many talents, but I hand it over to you, Kakani, and take it away.
Well, thank you so much, Tierney. And, you know, I'm so excited to be able to be included as part of this event. And really, you know, my lab and the people I work with, we're just having a blast doing what we're doing. And so I'm just excited to be able to share that with a much broader audience today. And so if I share my screen, let's go. So like Tierney had said earlier, my background is interestingly not in marine science or in the ocean. It's actually in aerospace engineering or in space. And in my lab, the theme of our lab is essentially searching for inspiration in the deep sea. And I'm going to explain why that's relevant and interesting. And although I'm talking about the deep sea today, I really did get my start by being inspired by space. I grew up wanting to be an astronaut, but in fact, I still want to be an astronaut. And I did submit my application to the astronaut pool a few months ago. So fingers crossed, we'll see what happens. But my entire life, I've been enamored by the search for life and what we could learn by that life once we find it. And astronauts, to me, you know, growing up, they were the ones doing that vital and important work in space. And I knew deep down that I wanted to do that as well. And so this is why I began my academic journey in STEM, and then later earned an undergraduate and graduate degrees in aeronautics and astronautics. And at some point in graduate school, I stopped looking for life in space. Oops, apologies. Tierney, do you see a video playing?
Yeah, it's playing.
Oh, okay, it doesn't play on my computer. I stopped looking for life in space and started looking at life in the ocean. And so the video you're seeing does not show a star field, but this is what the ocean looks like at 500 meters deep here in Monterey Bay. And, you know, the stars, right, they are actually particles or detritus and dead organisms and plants, and which is effectively food for most of the biomass in the oceans. And while we haven't been able to find life yet outside of our planet, the oceans are teeming with life that we know very, very little about. And not only do I want to search for life, I want to be able to do this in one of the most remote, least explored places in our ocean. And this is an animation that was created by Alex Winchell at National Geographic on a project that he and I were working on. And what you see is the research vessel Rachel Carson. This is the vessel that Mbari, where I work, operates. And then on the left is the Statue of Liberty. And when we talk about the ocean, we often think or automatically think about the upper ocean. And what that essentially is stacking two statues of liberties on top of one another. But when we talk about the scale of the ocean, right, the ocean extends from the upper ocean down to the seafloor. And this region, the ocean's midwaters, is the area that spans between them. And the ocean's midwaters is actually the largest habitable ecosystem on the planet, about 93% of it. And so while it may be a dark and cold place with pressures that are incredibly high, this place is filled with animals and exciting discoveries where these animals have evolved over millions of years to survive in this dark, desolate place. And so I want to find and discover life, but I also want to understand it. And so now as a bioengineer, I want to understand the mechanisms that enable these animals to survive the ocean's midwaters. And it's these mechanisms, or the physics of life, that can eventually lead to engineering innovations from bio-inspired design. And let's see. And much like observations by birds or early thinkers and doers like Leonardo da Vinci has led to our modern but still evolving concept of manned flight, what could we learn from animals that we haven't even observed yet? And in other words, what can we be inspired by in the deep sea? And so here's the problem. How do we access these animals to enable quantitative characterization of their physics of life? And my lab, the Bioinspiration Lab, is dedicated to developing technologies that address this need. We approach bio-inspired design in the deep ocean using this framework. By combining innovative observation platforms and state-of-the-art instrumentation, we can provide access to novel study systems and enable deep-sea bio-inspired engineering design. And innovative platforms and instrumentation are really the tools that allow us to evaluate the physics of life, right? Provides the contextual information about how animals live in their natural environment. But with access to animals, therefore, gives us the source of inspiration. And the idea is that if we can combine these features, we can then lead to deep-sea bio-inspired engineering design, which could someday lead to engineering solutions for problems that we as a society face. And so I want to share with you a case study that we've undergone using underwater robotics, laser-based imaging and instrumentation that our lab developed, and an out-of-this-world animal that we can find in the ocean's midwaters, the giant larvation. And so this is actually a photo taken of me at the Monterey Bay Aquarium Research Institute in front of one of our remotely operated vehicles, the ROV Doc Ricketts. So at Mbari, I am a principal engineer, and to further, my interests in bio-inspired design, this bus-sized remotely operated vehicle, or ROV, is incredibly important to me in my work. And so remotely operated vehicles like this one are rated to operate at depths up to 4,000 meters. And they have a suite of cameras, instruments, and tools that enable observations, collections, as well as experimentation. And so you can see, at least from this photo, these really bright lights, anywhere from 6 to 10 of them, that provide illumination in the depths. But in addition to all of this instrumentation that is present on these vehicles, my lab developed an instrument called Deep PIV, which utilizes laser-based illumination to image particles, gelatinous animals, and mucus structures in the deep sea. The system is deployed from an ROV manipulator arm, which you can see in this image here. And by deploying this from a remotely operated vehicle manipulator arm, it enables a 6-degree of freedom position refinement by an ROV pilot or an operator of that manipulator arm. Now, Deep PIV, the PIV actually stands for particle image velocimetry, which may be a familiar term for those of you that might be in the aeronautics or astronautics field, because it's a very commonly used fluid flow diagnostics technique by engineers or researchers trying to understand how flow might be moving in through turbines, how wings may be impacted by different angles of attack. And so this particular technique, where movement of particles and scattering media in a transparent fluid is used as a proxy for fluid motion, this technique is commonly done in the laboratory, and we repackage the system so that we can do these quantitative measurements underwater. So we also learned through this process that laser sheet illumination can also be used for 3D scanning. And I will show you some of these exciting results that were just published in Nature, gosh, I think like a month ago. But rated to 4,000 meters, we can use this quantitative imaging device to study the physics of life anywhere from the sea surface down to that depth rating. And so now let me tell you a little bit about giant larvaceans. Before I started this project several years ago, I had no idea what a larvation was, because I'd never taken like a basic marine invertebrate ecology course. And so what you're seeing is a giant larvation. I'm assuming you're at the clip where the animal is moving or beating its tail. And so that animal is shaped like a tadpole. But everything that's around that animal is a mucus structure that it secretes from cells that line its head. That mucus structure is used to filter food and particles from the water around the animal. And we call this mucus structure its house because the animal lives inside of it. And so while the animal might be about 10 centimeters long, its house can be up to 1 meter across. Oops. Let me make sure I'm in the right view. Here we go. And these animals, we believe these houses last for about a day. These animals will abandon the houses, and these structures that are rich in particles and carbon will sink rapidly to the bottom of the ocean and play an incredibly important role in the sequestration of carbon from near surface waters right down to the seafloor. And so I'm not a marine biologist, but an engineer. And I had no idea that something like this existed. I mean, how is this animal able to build something out of mucus? And why? How? How does this function? I had so many questions. And so we saw the deployment of Deep PIV as an incredible opportunity to try and answer some of these questions that have, really been asked by marine biologists for decades. And so the way this works is by using remotely operated vehicles, which are tethered vehicles or platforms connected to, a ship. And so both the ship's crew and the ROV pilots have to work completely in concert with each other to ensure that the vehicle is able to navigate to and observe the target of interest. And so what in the case of, say, a giant larvation, where these animals live anywhere from 200 to 400 meters deep, that requires a lot of collaboration between a lot of different people on a research vessel. And then once we come across an animal or a target of interest, what will happen is that, an ROV pilot will position the vehicle on an animal And then we will turn on our instrumentation to do some visualizations. But I want to point out the challenges at trying to do these kinds of measurements, where an ROV pilot who's sitting, at the surface on a ship, trying to maneuver and position a vehicle that can weigh about 11 tons, be able to, or 6.5 tons, I'm sorry, be able to position this vehicle on an animal that's 200 to 400 meters deep, maneuvering a laser imaging device with a sheet of light that's about 1 millimeter across on an animal that's about 2 centimeters wide, and then hold that position for a period of time while we do these quantitative measurements. And so this is a result of that collaboration with multiple individuals. So remember, we're using laser sheet illumination. So the only thing that's lit up is whatever is inside that laser sheet. So you can think of this as like an X-ray where you get to see inside features. And so I want to point out the animal's head, which we call the trunk, the animal's tail, and then that feature, those wisps that you see around that animal, that is actually a cross section of the inner filter, or that mucus structure that the animal lives inside. And if I play this video, and I hope you all get a chance to see this, you'll notice that as the animal isn't beating its tail, you'll see movement in particles. This is ambient suspended particulate that is just present in the water column. And as the animal starts pumping its tail, what you'll see is particles moving along down the tail, and then enter into the chambers of the mucus house. And so it's by the motion of these particles, right? We use it as a proxy for fluid motion. We can directly measure the filtration rates, or how much water these animals are processing in a period of time. And so based on some really incredible work by earlier researchers, Mary Silver, and among other people, we actually using scuba to observe similar animals, their estimates of how much water these animals are processing, on the come up to about 10 liters per hour. But by using our instrumentation that we developed, what we found instead was that on average, these animals are filtering at least 4 times more, but then in some cases, nearly an order of magnitude more. And if we scale that up to the population, and because Monterey Bay is one of the most heavily instrumented places in the ocean, the fact is we have incredible data over the past 30 years that gives us a sense of how midwater communities and populations might be changing over time. And so if we scaled our filtration rates up to the number of giant larvaceans that we can find in Monterey Bay, that would lead to these animals having the capability of filtering about 500 Olympic-sized swimming pools per hour, or filtering their entire depth range throughout Monterey Bay in less than 2 weeks. So pretty significant impact for animals that can be best case or as large as 10 centimeters in length. But here's the fun part. So when we deployed this instrument for the first time, we knew we could image particles. We knew we could image gelatinous features or gelatinous structures, but we didn't know how well we'd be able to deploy, or at least view, what's going on inside these gelatinous structures. And so after our very first deployments, we realized that much like a CT or MRI scan, we can use these laser-based illumination techniques to take images, consecutive images in a stack, and then reconstruct them into 3D models that we can then use to further investigate and understand what these mucus structures are and how they function. And so before I start this, I want to at least give you a sense of what you're about to see. So this is the laser sheet imaging device, DPIV. The ROV pilot has fixed or positioned the instrument in one place and is essentially moving the vehicle forward and back to capture these images that you're about to see. So Tierney, are you seeing the scan right now?
Yes, actually, we saw the beautiful scan, and now we're into the 3D scan.
Oh, okay, so you did see it.
You did see it. Yeah,
and so what we can do with these images And what we can do with this stack of information is we can reconstruct models, not only getting exterior views of what these models or these mucus structures look like, but actually unprecedented internal views of all of these different features, including what you're probably seeing now, these finger-like chambers that are emanating from these central chambers that all wind up meeting in one place near the animal's mouth. And so this whole process, and this, what is amazing to me, right, is because of our access to the ocean, right? We were using innovative observation platforms like like remotely operated vehicles. And by developing one instrument, in this case, deep PIV, it completely changes and opens up opportunities for how we can view animals in their natural environment in a completely different way. And we've applied that with midwater animals, including giant larvaceans. But for me as a bioengineer, has been understanding where all of this effort has led to. And in fact, there's been a number of different researchers that are partnering with our lab, interested in looking at a whole multitude of different science related questions. One in particular, I think this community would be interested in, is the fact that these animals create rudiments made out of mucus that they secrete on their heads, on the order of about 1 millimeter in size. But once these animals then expand these structures in, much like a balloon, and we've observed these animals only expanding their houses in midwater about 3 times in the history of Monterey Bay Aquarium Research Institute. But what's amazing is that these structures can go from 1 millimeter in size up to 1 meter in size. And so is that something that we can reverse engineer, perhaps learn how to do to deploy, incredible unfolding structures that humans can eventually inhabit in space? And so this is just, to me, an example of all the possibilities that are open to us as we further introduce new ocean technologies to observe this place. And so a project that we are nearly completed, or nearly completed development, is the Mesobot. It's a robot to investigate the ocean interior. This is a huge collaboration between my institution, Woods Hole Oceanographic Institution, Stanford, and University of Texas, Rio Grande Valley. This particular vehicle is optimized for midwater operation using imagery and coupled with vehicle control algorithms to autonomously track animals and other targets in midwater for up to 24 hours. We're also working on coupling this vehicle and future vehicles with algorithms that give it intelligence. And so I think a lot of people have been talking about machine learning lately, but in particular our group is trying to spin up the data that MBARI has been collecting for the past 30 years. It's all video and image data that have been manually annotated and augmented by experts. And what we're trying to do is spin up that data to create a training set that will be open to the public so that we can use it to train algorithms to not only annotate things on the fly and be able to detect and classify targets and animals in real time, but essentially fully automate the search for life in our oceans, to not only discover animals that we come across, but also to learn something about them. And so I am very, very excited about the possibility of this working, as it can have significant impact on how we can increase access to the ocean's midwaters, but also, more importantly, what this could mean for deep sea bio-inspired design. And so as we expand our eyes underwater and develop technologies that essentially will enable us to achieve a persistent presence, to observe and discover life in our oceans, who knows what kind of solutions we may be able to come up with from future lines of inquiry. And it's an incredibly exciting time for ocean exploration as we look towards understanding, but also really, truly being able to observe and learn from all of these different animal systems that we come across. And so as we can understand how these systems work, I hope we will one day be able to answer this question about how will we be inspired by the deep sea. So thank you so much for the opportunity to share my thoughts on all of this. And thank you to my team, as well as all of our collaborators, for this incredible work.
Thank you so much, Kakani. I think you're doing a lot to inspire us about the deep sea with your work. It's just phenomenal. You know, it makes me think of the first expeditions, like the Challenger expeditions of the 1870s, that when they dragged their nets through the midwater, all they came up with was a net full of goo.
Right.
Little did they know the intricate lives, the amazing bioengineering, the amazing ecomechanics that you're now being able to reveal, working hand in manipulator, hand
Exactly.
with robotic tools. And so I think your work is such a beautiful example of a human presence working with a robotic presence to really open our eyes. And blow our minds when it comes to the life that's in the ocean that we're just now discovering. Yeah. Amazing, amazing. Well, stay with us because we want to open it up and we want to have a discussion at the end of this. But thank you so much for that, Kakani.
Thank you, Tierney. Thank you for the opportunity.
Okay. So our next speaker has a very enviable job, an amazing job. Her name is Rachel Scott Butler, and she's done a lot of things. She's worked as a dive instructor, a marine biologist, but now she and she was a researcher at the Beeb, just a researcher, but they saw immense talent. And they promoted her, and she has become part of the core production team for such amazing shows as Great Barrier Reef, Shark, IMAX film Oceans, Blue Planet 2, has spent over 600 hours underwater capturing those amazing scenes that are just brilliant in our minds. Rachel's the one in the water capturing those scenes. So it just gives me, and when we talk about being able to tell stories from the sea, Rachel's right there telling them with her camera. So Rachel, take it away.
Wow, thank you for that introduction, Tierney. I feel incredibly humbled to be amongst so many amazing women, and I have to say I'm completely blown away by Kakani's presentation. Then I had no idea that technology existed and that life existed in the midwater. It suddenly got my brain sparking, thinking, right, how do we go about filming that for our next series? Blue Planet springs to mind.
Yes, I think we need a whole Blue Planet midwater.
Absolutely, we could do a whole series on that.
Series just on the midwater.
Who knows what's out there? It's amazing. Right, I'm trying to share my screen with you so I can tell you some stories from the underwater world that I've experienced over the last 10 years or so. So if I go, here we go. Can you see that all right?
Yeah, we see you just have to move to the top part.
Oh, that's right. Okay. Sorry, guys. Okay.
Oh, yeah, that's at the end of it. Okay. Is that the first slide?
Is that the picture of us all in our rebreather?
Yes.
Do you know, straight away, it's interesting, isn't it? Because I'm the only woman there. I'd never really thought that before, but perhaps we could discuss that later. But I mean, you've just introduced me so wonderfully there. I'm very I don't think I could say it the same myself, but I've like you say, I've spent about 10 years now working in the wildlife film industry. Before that I was a marine biologist, and I've spent a few thousand hours underwater now. But my job at the NHU now as a producer director is to find new stories, explore new technologies, and find new storytelling formats. Really, I think I should just be talking to Kakani because she'll be able to do all that for me. And our role at the Natural History Unit in Bristol is we try to connect our audience with the natural world and to give the natural world a voice. I work with Sir David Attenborough, who's here on the right-hand side, and like you say, I've spent a few thousand hours underwater. So as well as finding stories, I'm an underwater director as well. So I help to film them. Now, when trying to connect our audiences with the wonders of the ocean, we can do that through showing the grandeur and the splendor of these amazing habitats and landscapes. But we can also do that through amazing animal stories. And I think what I hadn't realized before I started in this role was that it's not just about capturing animal behaviors on film, but actually there's characters and narratives and a lot of intelligence and sophistication going on in underwater creatures. 5 years ago, I was lucky enough to work on a series called Blue Planet 2. It came 16 years after the original series, The Blue Planet. And the reason that my bosses and the commissioners decided that it was time for a sequel was because of the wealth of scientific information that was out there and the new stories that had come through from amazing scientists and naturalists all over the world, kind of finding new animal behaviors. But also, as we've seen in the first two presentations, it's because the technology has improved so much now, and we're able to tell stories now that we simply wouldn't have been able to do 16 years before. So I wanted to share with you two of those stories, and I wanted to introduce you to a little fish called Percy, the tuskfish. He lives on the Great Barrier Reef, and it's funny because I was listening to Sylvia and to Alina earlier talking about when they met a moray eel, and they'd given the moray eel, I think, Puff the Green Giant or something. And it's funny, I thought it was silly at first as naming a fish. But actually, when you spend enough time underwater with these characters, you realize that they really do have little personalities. Percy lives on the Great Barrier Reef, and reefs are incredibly competitive environments. They hold more diversity of life than anywhere else underwater, as far as we know at the moment. And because of that, there's a huge amount of sophistication and a lot of competition down there. The analogy of a city always comes to mind when we think of coral reefs. Of course, we have the changing guard between the night and the day, and there are so many different thousands of tiny little niches, and each animal is specialized into that niche. And we were trying to find stories that really illustrated that point. And our researcher, Jodi Lomask, came across a scientific paper that described tool use in fish. And but we didn't know kind of how frequently it happened, where we could find it, or how we could film it. So she contacted one of her colleagues, Dr. Alex Vale, who lived on Lizard Island in the Great Barrier Reef. And he'd lived there and dived there for about 17 years and had never noticed tuskfish using tools. But when Jodi said, Alex, will you head out and go for a couple of dives and see if you can see this? Sure enough, he found a few orange spotted tuskfish. And sure enough, they went around trying to find little clams and other invertebrates. And when they found them, they would swim them back to coral anvils and crack those clams against the anvil to break the shell open and get to the meat inside. And so that was enough for us to commission a shoot out to the Great Barrier Reef. And Jodi and I went out with Alex and cameraman Roger Munns, and we met Percy. There were a few different fish that were using these anvils, a few different of the tuskfish, but we found Percy and we loved him because A, he wasn't camera shy at all, whereas some of the other fish, believe it or not, were, and B, he kept coming back to this particular kitchen. And we spent around 100 hours with him, and we got to know his daily commute. And we would follow him, finding clams. Sometimes he would use his little fin and he would blow the sand away and find clams that way. Other times he would follow goatfish, and they've got these chemosensory barbels under their chins, and he would hang behind the goatfish and wait for the goatfish to find a clam and then sneak in and steal it from him. But he'd always take them back to this particular kitchen, as David called it, where he would break it open. We called him Percy because he was incredibly persistent. He would smash that clam for up to half an hour before he got into it. So he was a pretty amazing little fish. Now, here's a few shots of us trying to film him. Now, I found this story particularly interesting because, we know about cetaceans being intelligent. There's quite a lot of scientific information about that. But for fish to be capable of pre-planning and then be able to put together this coordinated and precise behavior, that's really incredible to see in a fish. And beforehand, I thought fish, had 10-second memories. I didn't realize that they could have personalities and could be very quite sophisticated with their behavior. To be able to film Percy, we really needed to immerse ourselves in his world. To do that, we used technology very similar to the Aquababes 50 years ago. We used rebreather technology, where you can spend hours and hours and hours underwater and you produce no bubbles. And so you're kind of in stealth mode in their world. We also really benefited by the advancement of camera technology, because now, and only really in the last 10 years or so, big chip cameras have become light sensitive enough that they can take these camera scopes you can see here. And this is effectively 4 lenses stacked up, one on top of the other. And it allows you to get very close to an animal, but be able to film it with a lot of depth of field. So normally, if any macro photographers are listening, normally when you're filming in macro, you've got a very shallow depth of field. But using a scope lens means that you can see the whole picture. We can really get down on the level of a nudibranch or the level of Percy and really immerse ourselves in their world. And like I say before now, this wasn't possible, and Blue Planet 2 really pushed the use of this technology underwater. We flooded a fair few cameras and lenses at the start before we managed to get it fully waterproof. But by being there on his level, we were able to kind of give these fish the Hollywood treatment and be, if you're telling a story of a city, you want to be down on the roads of the city with your character, not on the top of the skyscrapers looking down. And so by spending 100 hours with him and with the right technology, we were able to tell his little story. And this is a bit of, I hope you can see that video there. This is just a few shots of what we were able to capture with the scope lenses. And I should say, if you head over to the BBC Earth YouTube channel, then you'll be able to see the sequence of Percy narrated by Sir David. So understandably, after spending a month filming Percy and a number of other incredible behaviours and animals on the Great Barrier Reef, we were devastated when the following year in 2016, the Great Barrier Reef had a mass bleaching event. And it happened again the following year in 2017. And it's happened for a 3rd time in those 5 years since we filmed Percy, just this year. A lot of the reefs and reefs around the world are now bleaching. And I think, sitting in my front room in England, this often feels like a very remote, it's very sad, but something that I'm not really affected by, or people are not really affected by. But like I said earlier, our aim at the NHU is to really try and connect our audiences with the natural world, and we can do that through telling stories such as Percy's. And hopefully then when you see that the reef is bleached, you care more about the characters that live on it, the inhabitants of the city. Now I could talk all day about how amazing fish are, but I thought I'd broaden this out and give my second example of sea lions. Sea lions eat fish, but it wasn't known that they could eat yellowfin tuna. We heard about this behavior from a fisherman in the Galapagos Islands who had been fishing around this particular little bay here in Isabella. And he'd seen that there was a lot of tuna carcasses in the shallows. And he said to our cameraman, Richard, in the Galapagos, he said that the sea lions were chasing them down. Well, sea lions are a lot slower than yellowfin tuna, and yellowfin tuna can get up to 400 pounds. And so Richard said, I think you're mistaken. Like, there hasn't, nobody's ever observed that. Nobody's ever scientifically recorded this. But he said, no, it's happening, and sent us a few pictures. And so again, we headed out to the Galapagos a few years ago. We camped on this remote beach for about 5-6 weeks. And sure enough, after a couple of days of sitting and waiting, we were able to see a sea lion chase down and land a yellowfin tuna. Now, it's really with technology like these drones that we were able to really figure out exactly what was going on. The sea lions weren't working by themselves. They were working as a team. And from the air, we were able to see that they'd chosen this particular bay because of the topography of the area. You can see the lighter blue here is shallower water. And what these clever sea lions were doing was chasing the tuna up through this ever shallow labyrinth and eventually landing them and pushing them clean out of the water. And after, again, several days, weeks of observing this behavior and capturing it on camera, we noticed that, again, there were characters. And this little guy on the right is called Tag Boy. And there were a couple of big males. And we got to know them. And we got to know that when a particular sea lion headed out, you could be sure to get your cameras ready because he was going to come in with tuna. When they worked as a team, there was one that pushed the tuna up forward. There were flankers. And there was a blocker that would stop them from going back out into deep water. And they really acted like a pride of lions. I'd never really thought about sea lions, about that name before, but actually they were acting like a group of lions, like a pride of lions hunting down these poor tuna in the area. And there was a whole ecosystem that had formed because of the presence of these tuna carcasses in the water. When you just spent time and observed, you could see tiny little fish, you could see blennies, you could see crabs. There were a lot of birds that came down and all benefited by these sea lions and this extraordinary behavior. It really was an amazing thing to behold, and it was great because we managed to film it on camera for the first time. And yet again, just this coordinated hunting and sophistication was really surprising. I think I've been incredibly lucky to see these kinds of events over the last 10 years. But I think what surprises me the most of these experiences is not the amazing diversity of life. It's not the incredible behaviors that we've managed to capture on film, but it is the sophistication of these animals. And that makes me feel empathy for them and feel connected to the oceans. And I hope that's also true for our audience. You just don't think about underwater critters having that intelligence like you do with animals on land. But because of this advancement in technology, we're able to kind of level the playing field and spend hundreds of hours underwater with them. And I hope that through telling these stories, we have newfound respect for them and are hopefully better stewards of the world that we share with them.
Oh, thank you so much, Rachel. I think the work that you're doing, giving voice to the voiceless, is really the way that we're going to reach into the hearts of people to see that fish is just much more than just a piece of protein on your plate. When you say you're having fish, you could be talking about something that has an evolutionary history that goes back 500 million years. And every fish has its own ecological role. So the work you're doing is really bringing that home to the masses. And I applaud you many times over. Well, in the few minutes we have, I'd like to bring back all the Alina, and maybe we could have a brief discussion just amongst ourselves, and to start that off, I'd like to just have perhaps have each one of you say what would be your biggest call to action, say, for the next, the next 50 years. We've had 50 years since Tektite, and we've seen all these advances that Alina has laid the groundwork for. We've seen this amazing technology that Kakani's group is developing to tell these stories with Rachel and her storytelling techniques and camera work. We're bringing these stories to the public. How do we get through? What's your biggest hope for the future? Do you want to start, Kakani? Do you want to
Sure. Sure, Tierney. And I think this response won't be a surprise to you. In fact, it was largely, I think, inspired by a discussion we had nearly a year ago in Ireland, which I feel like is one of the last times we've been face to face, despite living like only an hour or like 20 minutes away from each other. But, you know, and I think this is a really great opportunity to discuss this, you know, that, you know, last year was the big anniversary for the Apollo missions. And, you know, there was this question about, well, what will be the next moonshot, right? And when we say the term moonshot, that means something really specific, right? That meant going to the moon. But now I think from, you know, more of this pop culture sense, that term means, you know, what's the next big thing that, you know, we as a society or humanity is going to work together and collaborate on and achieve and really change the way we, you know, experience our world, let's say. And for me, like, I feel really, really strongly about how, you know, the next moonshot should be us documenting all the life on this planet. Mm-hmm. And there's so many reasons to do that. I mean, it's not enough just to document it, right? You know, there's a lot we need to do in order to understand it. But there's all this research now showing that, you know, you can find life in deep sea sediments or, you know, in the crust of our planet. You can find life in the atmosphere or in clouds. And really, most people don't know that. And so if we really care, I mean, you know, the word sustainability might evoke different things to different people, but the fact is, like, we really want to take care of and nurture our home and, you know, really achieve sustainable living. You know, the first thing should perhaps be
knowing who we share the planet.
Know what we're sharing our planet with. And so for me, anyways, that I feel really strongly that should be something we focus our efforts on, especially
when we're losing so much in the so rapidly. Mm-hmm. Alina, what do you think?
Well, I'm a bit older than you guys. I've seen the world changed around me over my 74 years on Earth and the 50 years since Tektite. I'm very pessimistic about the future. I don't know what 50 years from now is going to look like. I don't have a good feeling about it. You know, when we did Tektite, there were somewhere around 2.5 billion to maybe 3 billion people on Earth. And there was a lot more stuff out there. And everybody talks about biodiversity and the loss of biodiversity and the sixth extinction and the Anthropocene. So I think that if we want to save the things we love, we're going to have to do open discussions about how do we stop human population growth before we eat everything, cut every tree down, kill everything in sight to make more space for humans. I mean, that is the sad reality from my point of view. And, you know, the younger people are going to have to figure out how to deal with this, right? It's just, it's going to be your Earth to live on when I'm gone. I have a 5-year-old granddaughter. I worry about what her life is she ever going to see any of the things that I saw during my years traveling and exploring. I love wildlife and everything I read, and maybe I participate with too many environmental organizations who always send me very depressing news about the elephants. You know, what we're doing on land is awful. So I don't know how do we get people to care about that and to shift the conversation, because we can explore and document, you know, on coral reefs. We've been documenting very well the loss of the coral reefs. And people keep trying to like grow little pieces of coral to glue them back, but the same factors that killed the earlier corals is going to kill the new corals. So, and climate change, everybody focuses on fossil fuels, but they don't realize that if we just change everything to renewable energy, but the number of people continue to increase, and the things that people do with energy, regardless of the source of the energy, continues to be the same, life as usual, we're still going to destroy things. So I mean, I hate to add that pessimistic point of view. You know, so how can we educate people with comedy or storytelling? We did a little comedy sketch. We didn't have time to show it, that tries to show the plight of the corals and how our instrument potentially, you know, could help in their conservation and saving. You know, would be the goal, but I don't know.
Yeah, I hear you. I hear you, Alina. And I urge people to go to the Kiss Me website and check out Alina's sketch that she put together, which you also are very adept at using medical metaphors to get across this idea that you know, we have to think of the earth as a patient like ourselves that's sick. And I think those kinds of metaphors are really powerful in terms of getting the message through. Rachel, with the work that you've been doing with the BBC and the massive impact of series like Blue Planet 2, are you seeing from the, you know, sort of how the series has done, the people who are tuning in and the different ways of tuning in, is that giving you cause for hope? Or as you see more and more, you're able to reach more people with all these different platforms that are available? How do you, from that perspective, how do you feel?
I completely see why you're pessimistic, Alina, because, you know, since we filmed Percy, like I say, his world was bleached 3 times. But at the same time, I think, you know, my role and our role as filmmakers is to try and connect people and try and get them to care. You know, and it's not just showing them how beautiful these worlds are, but it's saying, hey, there's a fish here that's really clever. You know, do you not want to save this world? And I think that kind of storytelling, you know, with series like Planet Earth, we told a bit more of a kind of what we call the God approach. You step back and you just show how beautiful the world is. And now we're kind of moving more into telling like really telling stories and telling stories of characters underwater. And I hope that gets people to care more about the natural world. And certainly in Britain, after Blue Planet 2 went out, it was very popular over here because we don't have so many TV channels, and there was a real, a genuine, tangible reduction in the amount of ocean plastic, or the amount of plastic, sorry, that we were putting in the oceans. Beach cleanups were improving by 50%, and people were really moving to using reusable bottles and using paper straws, and it's something very tangible that anyone and everyone can do, something like plastic. But I think I was so inspired by both of your presentations, and I think actually that the future is in the technology. Because like you were saying, Kakani, if we can follow that amazing huge invertebrate that blows not bubble up that big, I mean, what can we do with reusable energy, and what can we do with batteries? And I feel hopeful that, you know, my phone, you know, 10 years ago we had these big blocks, and now you've got a computer in your phone, and we have these huge cameras that can capture bioluminescence underwater. If we can just harness that technology and live more sustainably on our planet, then that gives me hope.
Yeah.
I'll add one little thing. I went to a film festival that was part of Ocean Week at the U.S. Capitol back 2005. Frank Capra Jr. was one of the filmmakers that was there, and a filmmaker by the name of Rob Taylor. I don't know if you know him. He specializes in marine mammal stuff, and he does incredibly beautiful things of dolphins and whales, and oh my gosh, you just want to jump in the water and see them. And he was at one film festival, and somebody came after him afterwards and said, you know, as long as you show all these beautiful scenes, everybody is going to, in the back of their mind, think that everything's okay. Everything's just as good as it's always been. And he thought about that. So, and this was before the U.S. Congress, he showed, everybody was in tears. He showed the baby seal slaughter up of Newfoundland, and these people bludgeoning these little cute little things and puddles of blood and everything else. And, you know, it made everybody want to call Canada and say, now, will you please stop this massacre? You know, this is awful. And you know, there was total silence because people were so impacted by what they saw. And then he said, okay, now that everybody's depressed, let me show you 3 minutes of beautiful, you know, whales jumping and everything else. So I think we have to be careful not to make everything look hunky-dory, because it's not. Right. So how do you introduce into your message that things are going the wrong direction? And if we want to save all these beautiful creatures, that now that you know that they're there, don't you want to have them be there forever? then we have to do something differently the way we live on Earth.
Yeah, and you're totally right. We need to tell those stories. We need to film bleaching as well as the amazing animal characters. And I'm working on Frozen Planet 2 at the moment, and I'm making a film about the Arctic, and we are really not shying away from the devastation that's happening up there at the same time as the amazing magical life that exists. And I think the combination of the two, the awe and wonder and the magic and the incredible characters, but also the destruction and the depressing scenes we're seeing will hopefully kind of kickstart people into wanting to do something to protect these beautiful, fragile worlds.
Yes. Well, I feel your pain, and I see the tears in your eyes, Alina. I think we all feel that.
I'm an easy crier.
But it's real, and we should experience it, and we should action on it. And so I would just like to thank all of you for this really moving panel. We have hope. We have our jobs cut out for us as storytellers, as technology developers, as stewards of the planet, and keep fighting the good fight, you guys. Yes, we'll make sure that everyone has links to your work, and we'll keep following everything you're creating. So thank you so much. And
Thank you, Judy, for organizing this.
Yes, no, it's my pleasure.
It's been wonderful. Thank you.
Okay, so we are going to move right on to our next panel, and this is it's been said. Ursula Le Guin observed that one of the functions of art is to give people the words to know their own experience. Storytelling is a tool for knowing who we are and what we want. And our last panel, we heard from Rachel and Alina and Kakani about stories, and now we're going to take a dive into into creative, powerful storytelling of the natural world by entering the world of dance through the work of Jodi Lomask, who is the founder and director of the renowned San Francisco dance troupe Capacitor. Sylvia, hi Jodi.
Hi everyone.
Sylvia Earle and I collaborated with you and your talented troupe on a performance entitled Okeanos a few years ago, and let's give a little taste of what that performance was like.
For sure.
Okay, and then we'll come back.
All living things are sea creatures, including humans. When you think about it, imagine Earth without an ocean. Imagine life without an ocean. The single non-negotiable thing that life requires is water. 97% is in the ocean. Take away the ocean. You take away life.
If you can sneak onto the reef and find a quiet hiding place. You can watch the whole seafloor come alive with hidden creatures.
Yeah, thank you, Jodi, for that highlights reel. That's wonderful.
Well, so I guess I should tell you a little bit about what I do and what Capacitor does. So what you saw there was one of our projects. I'm a creative director and a sculptor, choreographer, filmmaker, director person. I run a creative services agency, Capacitor, and we often work with scientists to create shows about the natural world. And I reached out to Tierney and to Sylvia Earle when we were developing the ideas for a new project about the ocean. And I feel like we were super lucky to get their time and their input on creating something that was a kind of celebration of ocean life, but it did reference and talk about the challenges that we're dealing with in ocean health.
Yes, and working with you was really sort of an eye-opener for me on a bunch of different fronts. I think what drew me to your work was this fact that when you watch dance, you sort of embody you sort of go along with the dancer. You're dancing with them in your mind.
Yeah.
And you did a piece on the rainforests in Costa Rica, and it was the first time I'd viscerally felt what it must be like to be a jungle plant fighting for space and light. And I thought, when I saw that, what if you could do that with an ocean creature, and you could embody an ocean creature in that way?
Yeah, so it takes we take a lot of time in our creative process. There's something called the Capacitor Lab we do for 6 months before we even attempt to make a show like this. And monthly we meet with scientists to talk about what they do, what they're studying, what they're passionate about. And the entire creative team participates. That means the costume designer, the composer, the performers, and it influences the sculptures that I design and the movements I choreograph. And basically the scientists become advisors to the entire creative team. And what's awesome about this process is it unifies the entire creative team. If you're gonna work with 30 creative people on a project, everybody's coming from a different place. Everyone has different experiences, and not everybody knows everything there is to know about whatever we're trying to create our show about. So in those 6 months, meeting with Tierney, meeting with Sylvia, meeting with other people at the Cal Academy of Sciences about the ocean, we were able to go as deeply as possible into the subject matter. Plus, I encouraged everybody to dive, to surf, to spend.
I know, I took you guys down to Point Lobos, and we were diving together. So.
Oh my goodness, that was so intense.
On a very cold day.
Yeah.
Yeah.
I got tangled in the kelp. It was really memorable. So the influence that the scientists have on the creative process is really obvious to me and everybody involved in our project. And in Okeanos, we went even a step further, and we integrated the voices of Tierney and Sylvia into our sound score, which we had never actually done. And I felt really great about that. I feel like we wanted to move people emotionally and help them connect to ocean life. But we also really wanted them to bring home some key information that might affect their choices.
Right. And we've talked about that idea of being sort of too on the nose, but finding that sort of sweet spot of abstraction as well through the artistic process. Yeah.
So how do you create great art that also educates the audience in a way that's not too
pedantic
or something? Mm-hmm. But I am curious, Tierney, how this process affected you, because for us it was really super rich, and we owe so much to you scientists, and I'm wondering what it was like on your end.
Yeah, well, as a scientist, you're sort of always thinking in these repeatable, methodical, logical ways, and that's what, when you're doing data collection, you have to have your methodology, you have to stick to your protocol, and but that's not the way the human mind always operates. We're highly emotional creatures, and sometimes irrational, and things come into us subliminally, affect our decision making, and working with you and your group, that really drove the point home. This, that messaging just through science is not going to get through to it'll reach some people, but the masses, we know we need to really pull out our creative stops to be able to message, because we feel things differently. I think Rachel Carson said it's not so half important to know as to feel. Mm-hmm. And so that pervade, really, that became much more of a powerful force in my life and in my mind as to how I looked at my science. And it actually made me turn my science to sort of delve deeper into the brain and understand how beauty and nature imagery affect our brain processes. And so I have research projects in that domain now.
Yeah.
So it was transformative, my work with you, because it really opened my eyes to the ways in which we receive information and what affects us, and then what causes us to act. Yeah.
It's interesting
but
that
maybe you could show enough.
Well, it's just interesting to see you draw those projects together, because I'm aware of that neuroscience work you're doing, and I hadn't really connected it to Okeanos. And in our in Capacitor's work, we went from Okeanos to creating synaptic motion, which was a collaboration with neuroscientists talking about creativity in the brain. And I think it's partly because when you're thinking deeply about environmental causes and how people make their decisions, and you learn that it's really, people can know all the statistics in the world, but we make our real decisions based on our emotions. It does make you think about, well, what's going on in our brains.
Yeah. So maybe we could show another quick clip about sort of two different ways of the storytelling. Yeah.
One of the creatures of the sea that I would really like to be is an octopus. Just imagine being an octopus. Having your whole body be like Rubber Man, that you could stretch your arm and it just keeps going and going and going, and you could go into the tiniest places by shrinking your body down to a fraction of its inflated size, or you could pump yourself up to be something that looks quite remarkable.
Hi. So that you really start to feel like what it is like to be an octopus. Working with Inka was really wonderful. Actually talking to her about all the bits and pieces, and I saw her interpretation change after speaking with her.
Yeah, deep into her performance a lot. She became quite obsessed with learning about the octopus, and I think that was able. It did a number of things. It made the performance much stronger. It made it better art, and when we're trying to communicate about the preciousness of ocean life, I think that came through much better. So the art as a tool for conservation was better in that regard. So yeah, I think with all of this work, we're just trying to tell the deepest story possible. We're trying to get into the experience of ocean life and not create a show of let's dress up like an animal and try to look like the animal, but how do we access what we imagine the feeling of being that creature could be? And if we can strike that deeper chord within ourselves, maybe we can with the audience, and they'll go there with us, and they'll feel the ocean in a different way, and then make the decisions based on those feelings that affect ocean health.
Yeah, I totally agree that embodiment. As a scientist, it's verboten to anthropomorphize an animal, but when you're messaging conservation, that is one of your most powerful leverage arms, one of your most powerful messaging tools. And certainly, I think art is what makes us human, but it's also what makes us humane. So.
Yeah, and it's often a question how abstract to go or how clear to make the messaging. And so in the example you just saw, it was a little bit more of an abstract interpretation of the octopus in a way. She was really just trying to get the feeling of the articulation and the independence of the limbs. But in our final video, we were trying to do something a little trickier, and that is try to share some really important information with the audience in a way that didn't take the potential for interpretation away from them. I feel that I guess I don't want to say too much before we see it.
But I think in the interest of time, we'll play that video, but then we'll close with that. So I'm gonna thank you, Jodi, for this conversation and for the work that you're doing. And then we're gonna close with Fish on a Line, and
thanks for having me.
We'll move on to the next.
Yeah.
But thank you so much, Jodi.
Yeah.
It's been such a pleasure.
Excellent.
Since 1950 and the advent of industrialized fishing, We've lost 90% of our big fishes. Our commercial fish stocks are being devastated. With millions of lines in the water, will we only stop after we've caught the very last fish?
Thank you so much for such an exquisite session. Jodi, Capacitor, Tierney, that was a beautiful segue into the next session. We're about to start a continuation of this journey to explore the theme of planetary habitation. We've brought together quite an interesting and diverse group of people, picking up on some of these themes about the forms of life, the signs of life that we see across our planet, and the many interdisciplinary perspectives that bring to bear in moments of discovery. These panelists were given a provocation, if you like. They were asked to consider the observations of Pierre Auger, the first president of the French Space Agency. And he had said, quite cheekily, that many scientists, he wrote, would like to call a sort of moratorium of science fiction and tell science fiction writers to hold it right there, to stop giving away what we're going to do. Because every time the scientists make a fresh breakthrough, the public are already used to the idea. They don't find new discoveries astonishing anymore, and that's a pity. Is there really a future for space exploration, he asks, or should we see the 1960s as a space oddity? I'm about to introduce a number of speakers who are dealing with concepts that in some sense would almost seem science fiction. They make you question what reality is, that which cannot be seen and yet can be still discovered. The first speaker is a distinguished oceanographer and an astrobiologist. It's Professor Jodi Deming. She is holds the Carl Bantz Professorship at the University of Washington. Her life is now looking at the limits of viral and microbial life in polar regions, and she's asking a really unique question, and that is, does the ocean have memory? Here is a pre-recorded session with Jodi.
These people who started with the childhood idea to be a scientist or a marine biologist or an oceanographer. No, I didn't know what I wanted to do, but I played the piano a lot. and had a scholarship to go to college on that, and had always done well in life science, but ended up with a pretty minimal science degree when I graduated from college. And then I had to get work to support myself. And that's a long story, but I ended up in a research lab supported by NASA. This was in 1974, so it was just at the time of the Viking Mars mission. And the lab that hired me as a technician had developed a method for detecting microbial life on Mars, but that method had not been used on the mission. And so what they wanted was to take this developed methodology and have earthly applications. Right. And they wanted somebody who didn't have a lot of textbook knowledge, who would have an open mind to some novel ways of doing things. They hired me right away, much to my surprise, and basically threw me in the lab and said, here's what we want to see if you can figure out how to make it happen. And that was my first chance to be creative at the lab bench. And that was just spectacular for me, because as a pianist, I had always felt that I was performing someone else's creation. That was immature on my part at the time, because of course, someone who plays music is very creative, can be very creative. But at the time, I wanted to be the one who did the creative thing. And in science, I've found that designing an experiment and acquiring the data and creating new knowledge was terribly exciting for me. So that's the beginning where I said, okay, I'll go to graduate school. And so then it goes from there.
And did you feel like you even though you were in technical capacity with the lab, did you feel like the composer or the musician or the instrument?
Oh no, I felt like the composer.
Brilliant.
Because I was very fortunate, because I had two supervisors, and it was in their mindset to give me freedom to design the experiment. They didn't design it for me and just tell me to go do it.
Yes.
And that made all the difference.
Yes.
That made all the difference. So it's the mentors you have in life who make all the difference.
It is true. It is true. It's potentially both competence and confidence. When you have the confidence to break the rules, you can start to design the game for yourself.
That's very true.
So tell me about your work in the field. You, from there, you've gone out and you are still continuing to search for microbial life and to understand that in the field, in the wild.
Yes. So my dissertation work way back when was about the deep sea. And at the time, in the 70s, we knew about animal life from the bottom of the ocean that not only tolerated but required the high pressures and the conditions of the bottom of the sea. Yes. But we didn't have any confirmed examples of bacteria, the smallest living organisms that were adapted to the deep sea. And this made no sense to me, because they are the most adaptable and they've been on the planet the longest, so it made no sense to me. So my basic contribution at that level was to spend a lot of time at sea and sample the deep sea and provide evidence that these organisms do indeed exist, and get them in culture and bring them into the lab and be able to work with them that way. So I started out as a deep sea microbiologist.
And what kind of depth were you able to work from a vessel, presumably on a tether?
I was working shipboard. Yes. And sending instrumentation down to the deepest depths. I was working in the Puerto Rico Trench, which is 8,400 meters deep.
Yes.
It's the deepest part of the Atlantic. There are slightly deeper parts in the Pacific. But I was sending instrumentation down, and instrumentation that would bring samples back up without losing pressure.
Okay.
So they were pressure retaining equipments, but I personally wasn't yet diving. But after my dissertation work, hydrothermal vents were discovered on the seafloor. And this was a brand new way of thinking that the bottom of the sea could be hot, because up until that point, everything about it was cold as far as we knew. And so I was a specialist on pressure adaptations at the time, but pressure and low temperature. Now here came pressure and high temperature. So I worked at hydrothermal vents on hydrothermal vent questions for a number of years, and that's when I had dives in the Alvin and also in the French submersible, the Nautilus, along the East Pacific Rise off of Mexico and also off the coast here. And I was looking at high temperature, high pressure microorganisms that emerge from the subsea floor at hydrothermal vents. And that started me thinking about life elsewhere. And to make a long story short, no, I can't make a long story short.
No, please don't.
Well, while I was doing that work, I had the opportunity to take a short trip into the Arctic and saw the ice-covered Arctic Ocean for the first time, which was very, almost spiritual for me. And then the Galileo space mission started returning images of Europa, the moon to Jupiter, which is also ice-covered and has ice features on it that are very, very reminiscent of our Arctic Ocean. So I did a pivot. I did a major gear shift from the bottom of the ocean to the top of the planet, and have spent the rest of my career studying life in ice, in particular sea ice, which is a salty form of ice.
Yes.
And that's important because if there's salt in the ice, then you can have liquid brine that is colder than the freezing point, because the salt depresses the freezing point. And it's also important because the ocean on Europa is salty. Okay. So at least some of the ice overlying it is salty ice, with little pockets of brine in there that could be inhabitable.
Yes.
And I just said to myself, okay, our best shots for finding life elsewhere in the solar system are these ice-covered planets and moons. But we don't know much at all about how life exists in ice on this planet.
No.
So I then have spent, I guess, decades now pursuing that question. And it has many spin-offs. And I guess the most recent spin-off is the ocean memory spin-off. Each of these stages of my career can be connected to ocean memory, but the ice phase is particularly on my mind now, because when seawater freezes, it captures what was in the water at the time. It basically captures the memory of that moment.
Yes.
But that ice lasts for a long time.
Yes.
When it finally melts, those memories are released. And here, memories are in quotes. They can be thought of as the microbes that were in the seawater when it froze in and now come out when the ice melts. And in the meantime, they do a lot of interesting things within the architecture of that ice. And I'm very fascinated by the architecture of sea ice and how that provides both barriers and opportunities for the microbes inside of the ice to interact with each other. But maybe now I'm getting too specific.
No, I don't think you are. And I think I do have a question before I want to backtrack on a point, but you have already talked about going from the depths and bringing it right out to the top. And are you going right out to the noctilucent clouds, to the ice crystals on the outer mesosphere, and looking at that redistribution of potentially a salty ice crystal forming at the polar regions out way beyond?
Yes.
Do you consider the whole planet an ocean?
Yes. I've written several proposals to do exactly that type of research.
Oh, brilliant.
None of which have been funded.
Yet.
However, that doesn't get in my way. For example, 2 falls ago, members of my team were aboard the Swedish icebreaker called Odin.
Yes.
That was working near the North Pole And the overall goal of that project was to study the upper atmosphere, the troposphere as well as the upper atmosphere. And the proposal that I had written but was not funded was to look at microbes up in that atmospheric space and how they tolerate or behave under those conditions. So, fortunately, I was funded for a different project on that same ship, and we connected it to the atmosphere by the fact that microorganisms in the surface of the ocean and in the surface of sea ice can become airborne, can be aerosoled up, and are known to travel high into the atmosphere. And so the organisms that we were funded to study in and around the ice are the seedbed for what ends up above. So I like to think that we're still contributing to that question without having actually sampled up there yet.
Yes. And how different is it to start to be in situ, to be able to be under pressure, to be in a submersible, to be under pressure yourself, to be in the environment when you're, say, looking at thermal vents, when you're looking for extremophiles in those places, and similarly going to the Arctic itself, rather than sending simply an instrument, but being there. How does that, you've already spoken about it being quite a spiritual experience when you first went there. And I implicitly understand the power and the magnetism of the ocean and the awe that is genuine and overwhelmingly connecting us to the planet. How is that linked to the microbial world? How do you read that architecture?
Mm-hmm. So, first of all, microbes are not visible to us. No. You have to have a high-powered microscope to see them.
Yes.
Or you have to do some fancy chemistry or DNA sequencing to get information about them. So I will admit that a large part or a part of my thinking is to use imagination and intuition about what they may be doing.
Yes.
Okay, so the question about actually being in the environment, when you're in a submersible in the bottom of the ocean, yes, you are definitely awestruck, always, continuously, but you are not actually experiencing the environment.
No.
You are not under pressure.
No.
You're sitting basically in a small office with control panels lit all around you, and you're comfortable in your own atmospheric pressure.
Yes.
You're not so comfortable because the temperature is a little cool. But whereas when you go to the Arctic and you're in the ice, you feel that minus 40 degrees C.
Yes.
You hold the ice in your hands. You stand on it. You experience it directly. Of course, directly as a human being, but you're actually experiencing the environment that your target organisms are experiencing.
Yes.
And that was very enlivening for me. It sounds awful to say, more so than diving to the bottom of the ocean. But it was actually, because it put me in more direct contact with the organisms to almost think like they do. Sorry for that extrapolation. But that's the sincere difference for me between the two extreme environments. The one I can experience directly, and the other that I can see and be in, but I'm not really experiencing.
That's right. That's right. I do understand that implicitly, and I think that that intuition is really important for the conceptualization of the ocean memory more broadly, and the way that you're not only conceiving of the organism and its exchange, but the broader body of water and its transference of knowledge and its containment of knowledge and its passage of knowledge, if you like,
mm-hmm.
that is inherent. Do you, as a potential codex of the past and the future. Is it important to be able to decipher this codex in any particular way that we haven't yet achieved?
There are so many examples of how studying the ocean from the perspective of memory can really educate us. I'll give you one that's very relevant today, since we are in the midst of a pandemic.
Yes.
Okay, the coronavirus is an RNA virus. RNA viruses, the best information suggests that they evolved in the ocean. And their ability to jump from one host to the next evolved in the ocean and moved on to land when land animals developed and land became available for them to develop. And the RNA virus is the ancient lineages of the current coronavirus, not the current coronavirus itself, but the ancient lineages traveled with their hosts and jumped hosts. And there are papers written on this, that the coronavirus community right now, which is very medically oriented for very excellent reasons, but they haven't really explored that literature yet. And it might educate us in some ways that would be quite different than the medical perspective. Just to gain a new connection to the ocean. I think many people, especially people who don't live near the ocean or who don't have an affinity for water or the ocean, have maybe a struggle to think about the ocean or its relevance in life. And yet it is the dominant feature on our planet, and it controls so many things about our lives and nature, everything about our planet. So any of these connections that we can make through memory, I think are very important. Humans have memory. Every single one of us, unless we have an issue, know what memory is about. We hold memories, we lose memories, we recall memories, we make plans based on memories. Yes. We anticipate the future threats, future pleasures. It's all about memory. Memory is an integral part of human life. It's an integral part of all life, and all life in the ocean. Even if organisms like microbial invisible organisms, they do not have neuronal brains like we do, but they have ways of interacting that create and constitute memory. And that's something that I'm very intent on exploring in the last phase of my career.
For the next phase of your career? The existing phase of your career?
The current phase, okay, fair enough.
The continuing phase.
Fair enough.
I think when we talk about life patterns and fluid systems, I think it's all a continuum. We understand it is a flow.
Fair enough.
It is a broad system.
Yeah.
So if
fluid is a good word today. Everything is fluid.
It is. It is. If you had a call to action for the future, whether it's through ocean memory or exploration in general for the next 50 years, where do you think we need to head, and where do we need to prioritize?
Very tough questions.
It is, yes.
It's especially the action part is very tough, because I don't think that all of humanity is in sync with the ocean at the moment, and we don't know how to think and act about it. So my first priority is an educational priority.
Yes.
To help as many people as possible to find and experience a connection with the ocean, so they appreciate that we have to pay attention to what we're doing and how it affects the ocean. Yes. It's going to come back at us. It is coming back at us. And sometimes, unless you respect something, you don't know to take care.
Sure.
So I would like to find a way, and I think ocean memory is a way, for people of all walks of life, any walk of life, to find the connection to the ocean, to respect it, and therefore to learn how to change our behaviors so that our planet goes forward and our futures go forward.
Unmuted helps. That was an extraordinary interview with Professor Deming, who is going to join us shortly in a panel. The notion of life in the ocean, or the ocean itself having memory as a signature of life, as a brain for this planet, is truly exquisite. And we look forward to diving in and exploring that further, particularly the architectures of the water itself, whether it's an ice crystal or a water droplet or a microbial system within that. Our next speaker also dives in to look at signs of life. She too is concerned and experimenting with the protocols for living systems and how they too may disrupt our expectations about, or the lens through which we understand the architectural property of life and living systems. Professor Rachel Armstrong is working in experimental architecture at the School of Architecture, Planning and Landscape at Newcastle University in the UK. She's working on advanced concepts, bringing together almost art, science, technology, and biology to investigate new approaches to building materials and as what we might call living architectures. So it is my pleasure to, I guess, dive deep into a different world, another world, and how we can experience this beautiful ocean planet and perhaps the extremophiles beyond. Rachel, welcome. You're on mute as well. You've done exactly as what I have done.
I've just unmuted myself. Well, if you don't mind, I'll just share my screen because I'd like to share some little bodies with you, if that's okay. So if that's okay, I'm just sharing the screen now, and hopefully I can pick up my. Hopefully we're here. Can you see me? Can you see my screen?
Yes, we can see everything.
Brilliant. Okay.
That's it.
Okay, fantastic. So I'm going to introduce you to these little bodies. They're dynamic droplets. They're only just visible with the naked eye, and they're here to help us think through and along with fluids. Now, the reason I think this is important is because whether in the guts of the abyss, the warm little ponds of our planet's surface, or the icy oceans of Europa, life as we know it exists in relation to fluids. Now, taking a leap into a liquid reality may help us see, experience, and imagine these other worlds anew. For if we are to better inhabit this world, and if our presence is to catalyze flourishing within others, then an expanded range of frameworks are needed to robustly respond to and work with the different contexts that we'll encounter. Our contemporary society uses the idea of the machine to find solutions to any challenge. So first, we reduce it into a set of basic operations. We propose a course of remedial action based on the assembly of parts, the construction of a machine. Then we draw power from an external energy source to configure and instruct the machine in ways that provide us with ultimate control over the outcome. Now, to step outside of that mindset is incredibly hard, as culturally, historically, and academically, our habitual approach is based on a set of assumptions framed by a particular kind of terrestrial backdrop beyond which our existence is constrained. So if we're to step outside of our present context so that we wish our interventions have qualitatively different kinds of impacts, and if we want to set up radically new approaches for engaging with unfamiliar environments, then our understanding of reality needs to be recalibrated. Now, thinking fluidly through the properties of liquids is a way of generating concepts for imagining, feeling, and experiencing worlds differently. So I'm inviting you to step inside a liquid world by considering yourself already immersed in a medium that's always in motion, be it gases or fluids, in which you can freely move and breathe. Let yourself think into your surroundings to experience this liquid reality, not just in terms of its material properties, but also consider how you are being infused, transformed, and extended by your environments. Now let's observe what's happening to these liquid bodies on the screen and see if their relatings to the spaces around them resonates with your own conjurings of a liquid life. And if you watch carefully, many of these actions are reminiscent of what we might recognize as being typical of the things we think of as being alive. For the ancient Greek philosopher Heraclitus, flux and opposition are necessary for life. So now let's consider how the infiltrations, extensions, and transformations that we can see on the screen and that we may have experienced ourselves in our imaginations can be considered technologically, so that we can coordinate them in ways that orchestrate specific actions. Now, liquid technology is very different than the technology of the machine. It's modulated through the spatial and temporal distribution of fields and interfaces. It possesses agency of its own and affords participatory engagement rather than promising absolute control. Inviting us to think of the kind of work we value, liquid technology asks what other kind of mediated work is possible. In his book, Dirt, The Ecstatic Skin of the Earth, William Bryant Logan notes that an acre of soil produces a horsepower of work every day. This metabolic work is not directed towards moving big machinery of parts around, but instead performs the work of life, which results in the overall enlivening of a space. So if we are to settle other worlds, and if our presence there is to generate an overall enlivening, then the technologies that can assist and perform the work of life are critical. We're not there yet, but advances in molecular biology have provided new insights into the nature of the technology of life. And if we can consider their implications through broader frameworks than the machine, then new kinds of understanding of how matter becomes lively may be possible. The liquid bodies that you see on the screen are not biological. They are entirely artificial, since they do not exist spontaneously in nature, and their constitution is entirely alien, even according to our working definitions of life, where a living agent possesses a metabolism, a container, and information. But they operate in a manner that's both extremely simple and irreducibly complex. Each droplet is contained only by the interface between oil and water, does not have a formal membrane. Their information is not centrally organized through nucleic acids, but is distributed through the chemical composition of their bodies and is activated where the oil and alkali fields overlap to produce soap. Now, this very simple metabolism alters the surface tension at the droplet boundary, which produces movement. and leaves a soft deposit or skin or memory in its wake. When the alkali runs out, the droplets expire. So like viruses, these bodies are not alive, but they produce effects relevant to the living realm and may be considered a technology. Now, the technological work produced by dynamic droplets is recognizable as the work of life, and the coordinated actions we observe can be influenced through introducing different chemical fields. So, for example, alcohol makes these droplets incredibly lively. Their responsiveness, though, provides us with a visualization system and tool for thinking through, so that we can start to better imagine the actual potential and possibilities of working through and with liquids, which may be considered a form of liquid computing. Now, to compute is to count, so that we may sort, order, and value the world. Conventionally, numbers are associated with modern computing, but nature's counting takes place through the iterations of molecular events, the expression of a protein, the repeated structural transformation of a receptor at an interface, the blink of an eye, a breath. Historically, we've counted with our fingers and toes, or digits, but advances in science and technology mean these digits are becoming stranger. Characterized by the rise of digital computing, modern computing links counting with the flow of electrons as bits through electronic circuits. And at the turn of the millennium, a new kind of digit emerged called a qubit that could be used for quantum computing. Now, qubits take many physical forms, like atoms, ions, photons, and even the same individual electrons with digital systems. And like bits, qubits are also measured using a binary system of ones and zeros. But unlike a classical bit, qubits can be both a 1 and a 0 at the same time, and it gets even stranger. Because a qubit can be both a 1 and a 0 at the same time, and what is measured determines what a qubit's final output will be. Now, qubits are the equivalent to classical bits in modern computing with a twist, because they use the quantum properties, namely superposition and entanglement. Now, this means quantum computing operations can be carried out more quickly and use less energy than digital computers, and makes them really well suited to solving certain mathematical problems, like finding very large prime numbers. And when Ada Lovelace imagined how Charles Babbage's analytical engine might act upon other things beside number, the equivalent challenge for computational platforms is not to imagine how they may perform existing tasks better, like make better numbers, but how we can find completely new uses for their capabilities. And for this, we must be aware of the constraints of our current thinking. So both at a digital and quantum computing level, they're acting at a distance from matter. And to close the gap between symbol and effect, chemistry itself must be involved, where Catherine Brahick notes, life, when you boil it right down, is a flow of electrons. You eat sugars that have excess electrons, and you breathe in the oxygen that willingly takes them. Our cells break down the sugars, and the electrons flow through them in a complex set of chemical reactions until they're passed on to electron-hungry oxygen. Now, liquid computing provides the embodiment necessary for information in the counting process to be spatialized, so it becomes architectural, and activated through its contextualization. An embodied process, information can not only be transmitted, but so directly acted upon without an interlocutor. So no longer merely indicative of a process for downstream translation by another apparatus, liquid computing involves the actual processes of life. So chemistry provides an infrastructure for the continuous flow of electrons, which takes place through the specific electron transfer systems in biological organisms that enables this never-ending web of potential permutations. Hopping from atomic seat to seat, the loss or gain of an electron confers different chemical properties to molecules with different effects. So this diverse chemical carrier is called metabolism, and it brings an animating principle to computing with matter, whose rich physical effects set material semiotic apparatuses in motion. Now, liquid computation creates the condition for negotiated exchanges and even experiences between substrates, a phenomenon that Eva Hayward calls fingery eyes. So the data within sensuous matter queers notions of sense-making, sensual meaning, value, and action, and which can be reproduced through materially entangled modes of digital or fingery-eyed computation. So these digits are neither the modern abstractions that we've grown accustomed to, but nor are they dissociated from their environment, but like our own fingers, can feel, sense, touch, slide into each other as well as the surroundings to negotiate outcomes of new kinds of haptic matter, whose tactile, kinesthetic, and proprioceptive senses function by contiguity, contact, and resonance, and feel their way around a medium. Implicating the liquid body in the computational process, rather than alienating it, establishes semi-living interfaces, concentrations, intensities, gradients, and depositions to replace the semiotics of classical modern computing. Rather than performing mechanical work, such computations engender autonomous behaviors, peculiar sensitivities, and soft time-based constructions that enable multiple kinds of programmable sensual bodies to orient themselves around their worlds. Now, whilst liquids typically are imagined as amorphous masses, recognizing and developing their computational and technological abilities could provide us with ways of feeling our surroundings, even if we cannot touch them, so that we can appreciate and remember them. A liquid computing that works with embodied informatic agents can generate a range of novel outputs that are not produced by modern computing. We will call some of these living materials. Others are types of technology, and a very few of them may even one day acquire the full status of a living thing. Computing with liquids, especially environments that are different than the terrestrial surface, could have far-reaching effects. Perhaps we will be able to confer our bodies with new organs that safely metabolize toxic compounds, or help seed agents in barren spaces that will help build entirely new ecosystems. Perhaps we'll use these characteristics to derive usable amounts of electricity from our environment that runs through semiconductor fibers performing the work of life that we wear like skins. Perhaps we'll build different kinds of habitats that will feel our presence with their fingery eyes. And as they do, perhaps we'll extend a high five or recoil at their uncanny ability to always know what we're doing. As we venture into new worlds, within our own world and beyond it, our cosmic duty within the community of life is to fundamentally alter the legacy of humanity to date. Through liquid computing, new configurations of life become possible, where rather than colonizing and disrupting new terrains, we can develop approaches that are capable of transforming our activities of daily living into countless world-making actions. So those are my little beasties, and as I say, they're much bigger than microbes, which Jodi was just talking about. But they're incredibly strange, and you can. Well, I have certainly got lost in worlds following them around under light microscopes, and so they teach me a lot of things, even though they are not living. I can start to see relations, the importance of environment, how even non-living bodies form micro communities, and really raise questions about our assumptions about one life and what it takes in order to world a world into a condition where the things that we empathize as living are present and can be with us.
And there's an extraordinary talk, and you've captured so much. It's beautiful, from remembering and enlivening to the technicity of this kind of material interaction. One of the parts of that, when you're talking about agency and bodies and the semi-living having agency, you were really stepping into a challenging domain for us to really reconceptualize how we conceive of life itself. And you talk about worlding. For those who are not, don't have a cinematic background, or perhaps describe the process of worlding for us.
Yeah, so I mean, I'm very keen on this term. So rather than, let's say, to make spaces, to make a building, to construct an environment, the idea of worlding a world, I think, creates the possibility of thinking in a very great holistic way about, first of all, being present with the actions that you're putting into the world, staying with the trouble and the responsibility of them, and let's say growing and iterating various possibilities so that actually you're present with change, and that you are there when you make your interventions and you're part of that world. So you're not a distant author. You're not separate from the world. You're actually participating along, perhaps with non-human agents, maybe other people as well. But the idea that that in some ways there's an ecological quality to worlding that doesn't separate the human from the non-human, and also has an ethical dimension in which we are implicated, non-innocent, cannot stand back and say, well, it wasn't my generation. It kind of involves us, and it means that we're not waiting for a time to have the kinds of effects we want to see in the world. We're right there in the middle of it and trying to figure out how this works.
Brilliant. And one of the ways you do that is also by forecasting as well. So I know that you have taken a lot of these concepts and considered them not only with their application today, but perhaps even looking at interstellar travel, for example, how these innate properties as a whole could be utilized as a functional architecture, as a way of seeding or supporting the future possibility for the building blocks of life.
I think that's very interesting, because what we tend to want to do is to export phenomena from elsewhere into a language that we understand, rather than. And I was just so fascinated by the comment earlier by Jodi, which was that when you're in the bathyscape or the submarine, that you're not actually immersed in the pressure of the undersea world. But when you're in the the icy polar regions, you are. And I think there's something very rich about that presence. And I think it's really about checking ourselves to ensure that we're not doing that colonial thing where we're imposing what kind of existence we want to see and then getting very frustrated when it doesn't happen, but actually trying to feel our way into what is possible, and becoming involved with the decision making with that, and not just thinking that that's something that happens on our world, but it will be something that we really need to think about when we step into new spaces.
Yes, and enabling systems that have agency of their own without having to control them is really important. I'm going to come back to the two of you, and I'd love for both yourself and Jodi to talk through whether we are going to consider the ocean and its sentience in the future as a very, or recognize its sentience, I guess, when you speak so highly or strongly of the the living quality of memory being signature of life and how we attribute it to the ocean, it would be a natural conclusion that we should be considering the ocean a living system with the full rights that could be afforded there. And what would that mean if we did so? We have some other questions. We have people who are really eager to talk with you about other thoughts and possibilities when we get to the interaction or the panel between all of you. And I can see this is going to be a fantastic discussion. We have another perspective to bring to the table, and this is, again, from the other side of the world, from a different type of researcher, one who's working at the intersection of neuroscience, biology, astrobiology, and again, looking at both photosynthetic properties and biomaterial properties, particular to aquaponic systems, and how they might be applied to the future space missions and the future sustainable utilization in space. And one of these garments that we're going to be introduced to, I have worn, and it has some incredible material properties that make me think of both of Rachel's semi-living systems and the interrelationship between body-like membranes and systems. So it was with great pleasure I introduce you to Dr. Agatha Kolejesk, who is the director of the Analog Astronaut Training Center in Poland. She's also worked with advanced concepts at the European Space Agency, and she works in collaboration there on a number of projects, including the one that we are about to hear from. Joe, we have Agatha's presentation.
Good afternoon, everyone. I'm calling from the stratospheric mission. We are just about to fly our biological and technological experiments, our CubeSats, into the stratosphere. This is the photo from our previous missions, and that's how I look like this, because usually during the missions we wear our cloth to feel like a team. And today I have an honor to be a part of this amazing virtual event. I would like to express my huge gratitude for invitation to be a part of this unique event, the 50th anniversary of the Tektite 2nd Mission 6 all-woman Aquanaut crew. It's also an honor to speak to one of the most influential women in the world, Honorary Professor Jane Lubchenco, former NOAA administrator, Professor Pascal Ehrenfreud, my idol, considering astrobiology, Chair of the Executive Board, DLR, President of the IAF, Tektite 2nd Mission 6 Aquanauts, Dr. Sylvia Early, Dr. Alina Szmant, Dr. Anne Harley, Margaret Ann, Peggy, Lucas Bond. I would like to acknowledge the memory of Renee True and to acknowledge Tektite 1st and 2nd program managers, Aquanauts, Dr. James Woodell, Jim Miller, Dr. Edward Ed Clifton, and Dr. Ian Koblik. Now I would like to invite you to my presentation. I hope it will be interesting. If you have any questions, of course, I'm very open to answer. In this presentation, I would like to present you a novel, ecologically friendly technology for the synthesis and modification of kombucha-derived bacterial cellulose in order to produce textiles of desired physicochemical and mechanical properties. The procedure of manufacturing cellulose in the form of a stable hydrogel bacterial cellulose ensures the desired properties for the application of such a material, for example, in the textile industry. Particularly, I would like to present you my project, which we made commonly with scientists and artists. And this project was named Bacterial Cellulose for Cloth Production in Sea and Space using kombucha microbial consortium. It was commonly realized, performed by Analog Astronaut Training Center, my company, my previous company, Space Garden, Department of Physical Chemistry and Electrochemistry at Jagiellonian University in Krakow, Department of Design, Art School Krakow, and Department of Art, Pedagogical University, also in Krakow. Bacterial cellulose was obtained from a yeast bacteria kombucha culture. You can see on this slide solution with something inside. It looks like a cave, but it is not a cave. On the top you can see multi-layered bacterial cellulose, which looks mostly like pancake. And below you can have you can see the fraction of yeasts and several bacteria that are that they like mostly environment without oxygen, so they stay here. And this symbiotic consortium consists of about 30 microbial species. It is very well known as tea fungus. It can be drank, this solution. It is very healthy, some people say. Also, it has common name as Scooby. It likes to grow on the black tea. This is the most common medium and very cheap and easy to breed. We, in our projects, develop different kinds of media which are based on algae bioreactors and on wastes. So this is very interesting considering future environmental issues. The process of bacterial cellulose manufacturing and modification in this project was optimized in order to obtain a maximum recovery of raw materials, minimal energy consumption, and to ensure the use of only natural and renewable resources. So this was our main technical goal. The obtained materials were characterized in terms of their wettability, mechanical properties, and flame resistance. And moreover, the morphology and composition of the materials were determined by using scanning electron microscopy and infrared spectroscopy, respectively. Here on this slide, you can see that we tested various kinds of materials at various stages of production. So first, on the left up, you can see the raw material in electron microscopy. Then you can see how this material looks after lyophilization and then after coating. We use several coatings depending on what we want to achieve at the end. Of course, I would like to share with some beautiful images, electron microscopy images of acidic bacterium producing bacterial cellulose polymer. It's beautiful. And here in the magnification, you can see that all these cells are drowning in the net of polymer fibers. Here also you can see that these fibers are randomly located everywhere. This kind of morphology allows, from one side, elasticity, flexibility, but from other side, very strong material. Here you can see some result from our anti-resistance for flames, for high temperatures, and we got very nice results, as you can see here. So this material doesn't like to be in fire. It's because of water, of course, that is inside this material. Here again you can see the bacterial cellulose, but this time we see the experiment with water. First, in A, you see raw material with coating, very simple coating, and then B, material is dried material after incubation 1 week in fresh water. In sea case, you see the same material, but incubated in salt water for 7 days. So here especially you can see some incrustations, some diffusion of salts into the polymer fibers. You see the crystals of salts that are there. Of course, if we would put this material back into the water, everything will be not dry, but it will be flexible again. Because we wanted to produce textiles, of course, we tried to generate some experiments with human skin, with first answering the answer if this is healthy to have this kind of gloves, if there are no allergic responses. And our first experiment was to produce wristbands, which you can see here on the A, they are newly made. And then in B and C, we see the same wristbands used in 2-week analog simulation. This was lunar mission spectra in Poland. And these wristbands were used by analog astronauts during the whole time of the analog simulation in the habitat. So even taking the shower, they had these wristbands. We also tried T-shirts, but not made of the only kombucha material. We didn't want to do this first. We wanted to again analyze various areas of body, how they react contacting kombucha material. And this is a T-shirt also used in this Spectra mission by the commander, Sarah Jane Pell. Maybe I should say more general about why we are doing this. So probably all of you know that one of the latest ecological problems when it comes to the use of gloves is the issue of biodegradability. Although items of clothing are used for a relatively long period, even by several consecutive users, and they have to be regularly washed, which often results in the release of fibers into water. And these fibers are mainly synthetic, such are not biodegradable. In consequence, they are accumulated in aquatic organisms, which is devastating for the entire natural environment. Moreover, the dimensions of such fibers may reach even the nanoscale. Thus, they cannot be easily filtered out and may pollute drinking water and also accumulate in human or animal organisms. In addition, the fashion industry has become one of the major contributors to water pollution, carbon dioxide emission, and waste. Therefore, it's crucial to introduce ecologically friendly technologies into production of textiles and post-production treatment of fabrics, to use as an example. Yeah, here are our analog astronauts of Spectra mission. What is even more interesting about kombucha materials and materials, ecological clothing in general, is that issues of ecological clothing production and their biodegradability will soon affect not only the population living on our planet, but also extraterrestrial human colonies. The topic of gloves is a challenge for astronauts, and it will soon become crucial due to the increasing number of planned space travels. As you know, textiles that are sent to space are not recycled under such conditions and cannot be washed, since space washing machine does not exist yet. More importantly, water is highly limited in the outer space. So the ideal solution would be clothing based on natural fabrics, which would be obtained from sources that could be cultivated or grown even in the most challenging conditions, such as spaceships or underwater. Under ideal conditions, these materials could be biodegradable, and the thus obtained biomass could be returned to the circulation by being reused in bioreactors. That's what we really would like to achieve. And here are the some photos from Spectra mission showing that at least this project was very successful. There were no allergic responses on the skin of our analog astronauts. Also, wearing a t-shirt was okay. Maybe not. This was not a full, full experience of kombucha material, but we got feedback that this material was cooling down when it was hot, and it was warming up when it was cold. So it was very interesting result. What we do now, we try to create some intelligence, some projects that will really work and will be very precise and accurate and sustainable. So what we do now, we try to develop environmentally friendly bacterial cellulose 3D printing method for production of customized fabrics in isolated spaces. And of course, our technical objectives are the same. So to solve one of the largest global problems, synthetic materials biodegradability. And we want to achieve this by development of practical, sustainable, environmentally friendly, and low-cost bacterial cellulose 3D printer. We call it BC3D. We would like to design and integrate natural filaments cultivated in situ. So all filaments that will be used to produce this kind of clothing should be natural and biodegradable. And then we, of course, would like to increase the autonomy in isolated environments, such marine platforms, such spaceships in the future. So we have to, of course, struggle with several kind of physics, natural laws, but I think everything is possible. Then, of course, we need to elaborate affordability of bacterial cellulose applied for large-scale fabrics. So we would like to make a very small machine comparing to the size of clothing that this small machine can produce. Of course, it is of one side earning the space, and the second thing is to make it as simple as possible. And here you see the draft of how it will look like. The material will be grown in the sterile liquid tank, which you can see here on this slide. The tank will be not only sterile, but it will be controlled by several controllers, microcontrollers. There will be a special alert system in order to ensure that everything is safe and is working properly. About filaments, I already told you. There is also some problems now that we have with extrusion, with the process of printing. So we need to have first some scaffold, and then there will be bacterial cellulose growing on the scaffold. So basically the printing will be mainly the scaffold and seeding some acidic bacteria producing polymers. That's what we do now, and we work in collaborations with many universities now on this project, and I hope that in 2 years we will have something very interesting to show. I have at the end a surprise for all of you, because I'm a flight director of the lunar simulation mission Bright 2. I asked my crew living in isolation in the habitat in Poland to send you warm greetings. So here they are.
Congratulations to the Tektite 2 Mission 6 crew, and thank you for inspiring future generations. Warm greetings from the Bright Lunar Simulation in Poland, Mission 2nd crew.
Thank you so much, Agatha, and thank you so much to the Bright 1 crew from the Analog Astronaut Training Center in Poland. I believe that they have landed back on Earth, and tomorrow there is another stratospheric mission. I believe there's a launch occurring, and I'm not sure what that payload is. But thank you for making the deep connection to this exquisite kind of use of material properties. We've had some exchange already with the speakers behind the scene talking about biofilm and the material properties from the sea and of course, which have memory as well. And perhaps there is a great convergence of ideas happening here. Our final speaker, who we will hear from now before entering into conversation, is a planetary archaeologist, or what we call a space archaeologist. Dr. Alice Gorman is internationally renowned in what is essentially an emerging field of space archaeology. She's putting together the pieces from a practice that has commenced here on planet Earth, of course. She has a background working with the cultural heritage of the indigenous populations here in Australia, and working with both the Australian and the Torres Strait Island groups to look at spaces of heritage here with deep cultural consequence, and using that as the basis to look ahead, to look at our space heritage and in exploration, including things like space junk, planetary landing sites, and so on. But we're delighted that she joins us today for a really special interpretation of this provocation. And Alice is going to present a reading for us to contemplate. Welcome, Alice.
I've just been so riveted by all of the previous speakers in this session, and I'm so happy to be part of celebrating Tektite 50/50. So I'd like to start by acknowledging the Wiradjuri people of New South Wales, from whose land I'm currently speaking. I acknowledge the importance of their cultural heritage and offer respect to their elders, past, present, and emerging, and extend that respect to the traditional owners and elders of all the countries that our audience is listening from as well. So at Sarah's request, what I want to do now is read you a passage from my book, Dr. Space Junk versus the Universe, Archaeology and the Future, which is about the field of space archaeology. And this passage is one that I think picks up on some of the themes in the session we've just had, and perhaps some others in the conference as well. So this passage is called True Infinite. Hyperobjects are not like other objects. You can't see them in their entirety, but you can perceive them through their effects. Once you have realized that a range of disparate effects are the manifestations of the same massive thing, it has moved from its unnoticed place in the background to the foreground of your consciousness. Then it filters everything about how you view the world. Or, as the common saying goes, once seen, it cannot be unseen. The solar system is something we can conceptualize, but not touch or see in its entirety. Usually it feels stable, like the mechanical solar system model called an orrery, where tiny artificial planets run on their preordained orbits. But when you look at the solar system's deep history, you realize that we're just living in a lull between catastrophic events like planets colliding or asteroid strikes. One such event is thought to have brought the age of the dinosaurs to an end approximately 66 million years ago. Every year, astronomers locate more and more objects in the near-Earth region, and the possibility of another major asteroid strike no longer seems like a random aberration, but business as usual in the solar system. The next such event might not happen for 1,000 years, but equally, it might be next year. The chaotic, unstable solar system is a hyperobject that won't let us rest. The more you know about it, the more unsettling it becomes. Hyperobjects are not located in a specific and precise place, nor in a particular moment. They're huge and distributed over vast distances of space and time. A human might perceive a local instance or manifestation of a hyperobject, but this doesn't mean they can grasp the totality of it. Dark matter is a good example of this characteristic. We only know it's there because it seems to be missing in action. When all of the matter of the universe is taken into account, it turns out there's not enough of it to provide the gravitational force for the galaxies to hold together in the way that we observe. There has to be some other mass out there. Dark matter permeates every corner of the universe, but it's only known by this gravitational effect. Laniakea, the supercluster of 100,000 galaxies that the Milky Way is part of, is another example of massive distribution through time and space. The cluster is 520 million light years in size, a distance that no human can ever travel. It's not a permanent structure and will drift apart at some unimaginable time in the future, just like terrestrial continents. If you look up into the night sky of the southern hemisphere and find the constellations Norma and Triangulum Australe, then you're looking through to the center of the supercluster. Of course, you might have guessed it, the Milky Way is out on the edges. Because of the data that was used to define Laniakea, pictures of it look like a curved and luxuriant ostrich feather. The galaxies in the supercluster are not represented by dots, but by paths of movement, the direction that they're traveling in. When as a child, I looked up towards the band of the Milky Way, I was seeing a tendril of Laniakea with the full glorious structure lying light years behind it. We could even argue that the cultural landscape created by space junk in Earth orbit is a hyperobject. It's spread over an area far vaster than Earth's. No one image can show us what it looks like. A photograph of a section of night sky might show several satellites as points of light, but you can't see all 23,000 large objects at once, and you certainly can't see the minute particles. We have to estimate what's out there using multiple sources and predictions, and then use this data to make images and simulations that are more easily digested. This is a very archaeological way of looking at things, studying the traces of a hyperobject in order to piece together the bigger picture using multiple strands of evidence and techniques. The information that Voyager 1 and 2 gather in their instruments about the region of space they're currently traveling through is similar. The data they send back to Earth are interpreted by scientists to make conclusions about the complex structure of interstellar space. Space anthropologist Michael Oman Reagan argues that interstellar space is itself a hyperobject. If you think of it this way, he says, we encounter it at many different scales. It's not something remote and desolate and impossibly far away. It's close and intimate as well. The signs by which we recognize it in everyday life don't require instruments to read. Oman Reagan says we can perceive the traces of interstellar space in writings, like those of Carl Sagan and Herman Melville, and in the feelings that their words evoke. I've tried to read Moby Dick, Melville's classic novel about whaling in the 19th century. I really have, but I've never been able to make it past the first few pages. But I did find out what Michael meant. The novel is about Captain Ahab's quest to kill the white whale which injured him. One day, the main character Ishmael looks over the edge of the boat to see the whale lurking under the surface of the ocean. He thinks to himself, and I quote, "Is it that by its indefiniteness, it shadows forth the heartless voids and immensities of the universe, and thus stabs us from behind with the thought of annihilation when beholding the white depths of the Milky Way?" End quote. Interstellar space is hidden in the depths below the whale, whose milky white body only serves to accentuate the formless space in the dark sea. It's an as above, so below moment, in reverse. The stabbing from behind immediately made me recall a concept from Frank Herbert's Dune novels. This is ADAB, the demanding memory that comes upon you of itself. At some deep level, the void is always present just beyond the limits of memory and sense, until it forces itself into your consciousness. Plato had a similar concept in Anamnesis, the theory that all knowledge was really just remembering things that we already knew from an eternal existence. This isn't knowledge that involves physics and mathematics. It's emotional and psychological, and no less valid for that. If I can make another connection here, then I'm reminded of Freud's theory of the uncanny, the thing that is horrifying because it's so familiar, like the doppelganger, the white whale doubling as the Milky Way and calling the void forth into one's soul. It doesn't always have to be horrifying, though. The white whale might summon forth the oceanic feeling rather than the heartless void. This is a feeling of oneness with the universe that French writer and philosopher Romain Rolland, drawing on Hindu and Sanskrit mysticism, thought was the origin of the world's religions. He wrote about it in a famous letter to Sigmund Freud in 1927, and the idea unsettled Freud so much that it took him 2 years to reply, a fact which makes me feel a lot better about my out-of-control email inbox. The comparison of space to the ocean has been made many times by people such as Arthur C. Clarke, Carl Sagan, and space historian William E. Burroughs. Contemplation of the ocean can be a door to a transcendent experience, to a cosmic ineffable that expands the soul. This experience, though, seems to be temporary and evanescent. You're not in that moment all the time. Hyperobjects might be demanding and viscous in the way they shape your perceptions, but they're also forgettable. You glimpse them and they can be overwhelming, but they're hard to hold in your mind. You need something to recall them and make them stick. Something may jolt you out of the forgetfulness, like the re-entry of a piece of space junk, or an asteroid nearing Earth, or the plastic bag found at the bottom of the Mariana Trench. But you gradually slip out of your new way of thinking and back into the old familiar one. That's it. Thank you.
That was so beautiful. Thank you. And what an extraordinary encapsulation and provocation for all of us to respond to and to unite us. I'm going to ask Rachel Armstrong, Jodi Deming, and Agatha, I apologize, I can never pronounce your surname. Welcome, ladies, and thank you so much for your gift of both insight, sensitivity, and breadth of interdisciplinary knowledge that you bring to each of the questions that you're pursuing, and the way that you articulate them is extraordinary. There's been a multitude of comments and chats behind the scenes, and I think you've all made the connections that I hoped you would between you. Is there anyone who would like to start and make a response or question or a comment for each other?
I'll make an observation. I think one of the things that I really, really enjoyed about all the talks was the idea that these spaces aren't out there, they're very close, and its proximity, the body, and there's a kind of materialization that's running through, which I really love. And it's not a classic let's say a chemistry recipe, you know, like this planet is made of this stuff. But there's a kind of relatability to all the worlds that you've brought to us. And I think also very interestingly, a language that helps us make those sensuous connections with other worlds and others. I mean, I was just listening to Jodi, and I felt really attached to all those microbes, and same with Alice. I mean, hyper objects, yeah, I want my fingers in everything. And agates, so I want to be wearing your biofilms. So I think, so for me, there was something very sensuous, material, and very proximate, despite the vastness, despite the difference in scales, despite the difficulties, and it felt very relatable.
Yes. Terrific. Alice, you have, as an anthropologist, you are trying to make sense of a lot of this scale from that human perspective as well, as Rachel was talking. From your perspective, before Rachel and Jodi make a comment, do you think of the ocean as something that is almost sentient, that has agency, it has memory, that it is part of our cultural heritage, but that's the wrong word, but how do you relate to the ocean in all of this?
Oh, I thought you said something very interesting, Sarah, introducing something earlier on, because I actually don't personally feel any sort of strong connections to the ocean, perhaps because I grew up very far inland, and it was always this incredibly exotic thing. But shells for me were wondrous and mysterious. I used to collect shells, even though, you know, the possibility of getting them happened so rarely. But I think, so I'm thinking about a lot of what I've heard everybody talk about today, sort of in a solar system context, like where are the other oceans, where are the other ice particles, where are the other liquids doing this work? And liquids aren't always what we think of as a liquid, like soil behaves as a liquid. So something I was thinking of as Rachel was talking is about the work done in an archaeological deposit, which normally we think of as a layer of dirt, which it is, but it moves. It behaves like a liquid over a long period of time. It moves artifacts around. It breaks relationships between things in that deposit, which as archaeologists we're sort of trying to reconstruct. And something I've been thinking a lot about is the qualities or characteristics of the stuff that's in interplanetary and interstellar space. Is incredibly widely dispersed, you know, the least dense gas you could imagine, particles that are incredibly far distances apart, traveling at very high speeds. I'm trying to think of how to characterize the qualities of this environment and then characterize what are the human inputs and contributions to that. But listening to everyone this morning, I've been drawing inspiration. I mean, I know this is an ocean. You asked about the ocean, and this is not about ocean now, but space is compared to an ocean. I'm thinking about the interactions of all of those particles and the life they have of their own. When you put human stuff into it, and the sort of normal way of considering things, and this is probably similar to the ocean too, like you put stuff that somehow leaves no trace. You can remove it as if it had never been there. But it has been there, and it will always have been there. And what is left is something that is actually a new entity with qualities of its own. And everything I've been listening to this morning has just been inspiring me to try and think of more creative ways to talk about what that thing is.
Jodi, that's part of your work, and that is to think about creative ways about how to undertake those kinds of investigations with ocean memory and to bring that together. How have you responded to this collection of provocations and perspectives today?
Well, I see memory in everything now. Ever since I've been involved in the Ocean Memory Project, That's my frame of reference for everything. So I see it in Agatha's fabric creation, those biofilms that carry a memory of their creation and how that happened, how that was instigated bacterially, and how those strands carry forward. And I definitely see it in Rachel's liquid forms that are so microbial to watch. In fact, now I'm going to digress, but it raises the question of what is the definition of life, and how do we explore for it if we don't have a clear definition of it? For example, if we explore for life as we know it, if we explore for little microbes that we can very well characterize on this planet, well, what if we see forms that look like microbes or behave like microbes, but they are really Rachel's liquid forms that may or may not be identifiable as life forms? You talked about how eventually they might be characterized that way. So how do we go about this process of defining life, which is so essential to being able to search for it? What are we searching for? If we're searching for any possibility, wow. But if we're searching for something discrete as we know it, that's very limiting. So there's this whole range in between. How do we define life? That's inherent to our astrobiology program here at UW. That's a constant theme that our students are asked to wrestle with. But then I'd also like to comment to Alice about these spaces, these hyper objects. I can only think as far as our solar system, sorry. But there are multiple oceans in our solar system. And I think they may have a collective memory of their formation. And perhaps it's very archaeological in the sense that there's a layering in the solar system. Planets are layered out there, and there is a habitable zone that is identifiable in those strata, and ice-covered oceans fall in and near that zone. Okay, I should stop because I'm getting excited in my head.
You're meant to get excited. Agatha gets excited. You're also trying to look at the habitable zone and to prepare crews for that. How do you prepare crews, just like Jodi working with astrobiology, how do you prepare for discovery of things beyond things that we cannot see and things that we may not yet have a conceptual frame of reference for?
Well, we are trying to rise to breed some bioreactors with microbial life. Of course, kombucha, what I showed you before in the presentation, but also some algae, symbiotic consortia. So not only single algae species in one bioreactor, but we try to combine several algae because we see that they are more resistant to stress and they can sustain longer in isolated space. So together means stronger, I would say like this, and we can really physically observe this kind of symbiotic interactions in microbial life. We also try to elaborate optimal media for hydroponic systems in order to have nice fresh food in isolated spaces. And here again, we have several problems with microbial life, starting from biocontamination, how to prevent growing wrong bacteria, wrong fungi, and how to increase the health growth of what we really want to grow. So we struggle with several problems, but again, we are trying, and it is just by experiment what we can see, that adding some microbiomes into the medium, so some life to the medium, not only organic chemistry, microelements, nutrients, but also some life. We see that these media are much better in order to grow plants. So this is very interesting what we are now working on.
And a quick question, Agatha, do you see this space as an ocean as well?
Now, yes, after what I heard here, something opened in my brain, you know?
Well, an astronaut is a star sailor, yes? So that makes some sense. Yes.
I mean, definitely, especially in confined space, we can feel that everything is connected with each other. So one single change will induce several changes, you know, in all bodies, but also in the whole habitat. So we can observe by just standard environmental sensors that one small change will implicate several changes in larger scale. So now I can say that yes, all isolated spaces are similar to the ocean, to the processes that occur globally. That's amazing that we can rescale some processes.
Jodi, sorry.
Just something I'm thinking a lot about at the moment as well is the permanently shadowed regions, which are on the Moon, Mercury, and Ceres, where there are the water ice deposits that everyone's so keen to get to on the Moon. And I think there's a lot of stuff in what everyone's been saying about how to describe these. So they're just, at the moment, they're being described as a resource or a chemical composition. And I'm thinking about the shadows, the nature of the shadows that have enabled these deposits to be stuck there. But then these are things, in terms of the intrinsic value these water ice deposits might have in these locations, they are doing, in some way, they're doing work in the interaction or the, in fact, well, I don't know enough about it to know how ices interact, but you guys know more. They are doing some kind of work on their own, and they are capturing the state that, you know, that precise chemical composition of the time or the period of time over which they were laid down. So this is helping me kind of think about what are those, what are the intrinsic values of 2 billion-year-old ice deposits on other planets in the solar system. And as you were saying, Agatha, like a change in one part can affect a change in another, which is what you're seeing in the work that you're doing. And we don't know, we don't even know enough about what these deposits are in order to even begin to understand how a change like the NASA probe that is going to go and shine a light into shadows that have existed unbroken for 2 billion years. They'll shine a little light down onto the surface of the ice so that they can get data. So the easy thing is to think, well, that's not going to make any difference. It's a tiny, tiny, tiny little torch being shone down. But that's the beginning of this other process, the beginning of the information people need to mine this stuff. And what change does that, what happens when you break a 2 billion-year-old shadow and illuminate the surface of water ice that is laying there for that whole time, interacting only with cosmic rays and a few micrometeorites and bombardment by other particles. So yeah, I think there's a lot in what everyone has been saying that for me is really helpful in thinking about how to work out what this means.
Yeah, I think there's a story that I think we've kind of lost since around about Linnaeus's time, which is about, well, it's coming back now, but the story of life and its rocks. That there are more minerals on planet Earth because of the activity of life, and that the activity of minerals has actually shaped, geological, cycles and systems. So, carbon dioxide being trapped with the silica cycle, and but the fact that we have, many more minerals on this planet, and I think that you're absolutely right that kind of delicate relationship between environment and the figure, whatever that is, the patch of chemistry, the a catalysis that happens at an interface between one space and another. Those are all the very delicate things that may be disrupted in a way that makes a space change radically from what it was. And I also think that another thing for me that was quite interesting was the kind of de-anthropocentrization of the narratives. I love the idea of the ocean memory, and I think it's real. I think we have a very human-centered view of what intelligence is. I mean, but the idea that intelligence and memory could be highly distributed, like Alice's hyperobjects, it doesn't mean that because they're not all condensed in a little wet thing that we call a brain, that it's any less responsive and any less able to interpret and make decisions, even if those are molecular, about events and contingencies. So I think that one of the things that I think would be interesting is if we have an expanded view and we say what our preferences and prejudices, and we're kind of completely aware that this isn't empirically provable. However, if we're looking at these kinds of contexts and possibilities, then maybe we can think about things this way, and then we can set up experiments that help us negotiate those ideas and kind of interrogate them. I think if we have to reduce everything to empiricism, I think we miss stuff, and I think that's exactly what Jodi was saying about how do we define life, and there's a kind of relatability there that isn't empirical.
Mm-hmm. So I'd like to just explain briefly that ocean memory is a very broad concept. It's not just that the ocean itself holds memory, although there's a whole body of work on how water masses do hold memory, but there's also chemical memory in the ocean, different chemical reactions that trigger new processes, that recall old processes, that there's a lot going on chemically. And then there's the whole world of life in the ocean with different organisms and different ecosystems and communities encoding memory genetically or at the ecosystem level. So I wouldn't want you to think of ocean memory as being constrained in any way. It's also not just science, it's totally metaphor and any way that you want to think about things. We don't want it to be narrowly defined. We want it to be as broadly defined as possible, because metaphor can be so important. As Alice read there, that wonderful metaphor with the Moby Dick and the Milky Way, that was just fantastic. I wasn't familiar with that metaphor. Thank you for that. Sorry, now I'm excited again. I've lost my train of thought. Go for it. Rachel, what had you challenged me to address before I went off on my spiel?
No, I'm very, very interested in your definition of memory, and I think that's.
Oh yeah.
I think it's really, really interesting. The fact that there are multiple layers to it, so that they build on each other. So yes, there is water itself, the weird molecule that we're still fathoming, and then there are the mineral contaminants and the solutions and the pressure gradients, allowing layers of difference. The possibility of invisible interfaces we can't see with our eyes, but with different sensors, maybe we could taste them. And then the genetic elements and the elements of ecosystem. It's just beautiful the way that you've described that. It just feels incredibly rich, and you could see how something like that is incredibly complex and can tell you stuff.
Lots of stuff. And I remember what I wanted to refer to just before our session. There was this absolutely beautiful dance of the octopus. I don't know if any of you saw it, but so the octopus is obviously a marine organism, and it has a distributed form of memory. It has more neurons in its arms than we have in our brain, and each arm has a different memory. and it's very tactile memory. So one arm, like the dancer, is touching part of this ball that she immersed herself in, and another arm or her leg is touching another space. The different brains in the arms are remembering things differently. And the actual brain of the octopus is very small, but it does play the role of trying to assemble all those 8 different memories and take action. And this is a form of distributed memory that we could learn a lot about, because we only know about the single brain form of memory, or most people only know about that. If we could think about distributed memory and other ways of developing memory, that's very mind expanding.
We have touched briefly on this before in discussion. We have talked about the possibility of the body and the body of water having extended knowledge, but perhaps working through a choreography to extend the knowledge from the body to the body of water and the body of water back to the human. And what would that require? Does that require tapping into indigenous knowledge systems, to aquatic knowledge systems, where we become the soapy interactions of life, where we have the agency of, that chemical agency, we become enlivened and we become responsive to ourselves being part of this bigger system of, as a whole? And if so, what is the technicity that enables that? Because I think all of you have touched on, and this has been a great theme of the day, how do we enable people to develop a consciousness and awareness and a care for the ocean, because it has such a significant role to play in every living system on this planet. And in fact, from this session, we might say that it is the living system of this planet, that we are inseparable from it. If we are to conclude here, and we become the 8-armed octopus with a particular type of memory, and with remembered with a point of action, a tentacle that we could spread and share, what would be your call to action? What would be the memory that you would like to instill with people that encapsulates today's discussion for the next generation?
For me, it's this idea of worlding. I mean, I think the memory of the ocean and the hyper objects and the the microbial structuring, that they're incredibly complex, but we're involved with all this. And I think rather than thinking about sustainability, and I was listening to some talks earlier, and sustainability is still used as a word. I think for me, it's inviting a status quo. And I think for me, worlding emplaces us in the action of change itself, and gives us that responsibility and ethics of taking care and taking with. So that for me, worlding is the thing to go forward with.
Perfect. Jodi?
I can't really improve on that, except to again say that I think that memory is a beautiful way for humans to connect to the ocean. And once we all connect to it, and connect to it as fully as we can, then we will take care properly.
Beautiful, beautiful. Agatha?
Well, I think that what I think it would be important is to educate people that we are not alone, and no one is alone. We are all connected, and nothing happens without a reason. So it would be great if we, humanity, if human civilization, could understand more these all relations between the nature and us, because this is, I think, the crucial thing now in all progress of technology, which is kind of dangerous. So this conscious and this this feeling that we are not alone, that our decisions are not only our decisions.
Alice?
thinking about what everyone's spoken of today, I think a theme throughout this has been the intimacy of surfaces in contact with each other. And it's that intimacy that situates us in the world that we're living in. And I think paying attention to that embeddedness and embodiedness within the clothes we're wearing and the atmosphere we're interacting with and the water and the liquids that we engage with every day is maybe a useful way of thinking about the actions that we need to take, or thinking about how that world that we have created isn't static. I agree, Rachel, with the sustainability issue. I've been having a problem with that for a while, because I think people are happy to exist and let things go, and making ways we can connect with these much broader objects through these surfaces that we touch is perhaps a way of getting more thoughtfulness and hence more action and decisions about the kind of world we want in the future.
Beautiful. Beautiful. Well, thank you so much, all of you. I got some feedback there. We'll just try that. Thank you, Alice. I think that was your your loop there. Thank you so much for an exquisite discussion and all of your beautiful, thoughtful ideas. There are incredible metaphors there, but also some practical considerations for how we move forward. We're going to conclude this session. Thank all of our guests, but conclude this session with a short work by another artist, an artist who is a space artist and a radio telescope operator. And she has been working with the philosopher Frank White, who is the author of The Overview Effect. And together they developed a virtual reality piece where people were experiencing a virtual reality interpretation to evoke the notion of the overview effect. And they're working with neurologists and a team of radio astronomers. She was working to stage an event at the Dwingler radio telescope in the Netherlands and to send their brainwave transmission into space. This is Daniela de Poulis and her work Cogito in Space.
Space is really an extension of the body. Between the body and the outer space, there is just this membrane which divides each individual in a way. Space exploration for me is not about as an exploration, for example, of the unknown or as a colonial exploration, but it's really about exploring ourselves in this bigger dimension, which is also part of
Joe,
ourselves.
if we could just restart the video with the audio, please. You'll need to take
Probably
your
there's a-
headset out so the audio could play.
Space is really an extension of the body. Between the body and the outer space, there is just this membrane which divides each individual in a way. Space exploration for me is not about as an exploration, for example, of the unknown or as a colonial exploration, but it's really about exploring ourselves in this bigger dimension, which is also part of ourselves. Cogito is a project where we send brainwaves into space. We record them with a sophisticated device, and we convert it into a sound that can be transmitted into space in real time. We have 32 electrodes, and these 32 electrodes record the electrical signals across the entire brain and convey the signal into this box, which is a wireless device that transmits the signal to a receiver, which is connected to a computer. I wanted the scientific element of the project to be very clear, very strong. The neuroscientists really played a very important role. I really want the brain activity to be as best as it could. For the radio transmissions, of course, I work with radio astronomers, also radio operators. The language of radio and the language of neuroscience have nothing in common. We record one particular kind of brain activity, which is the response to a video in virtual reality, which is created around the idea of the overview effect, so the idea of seeing the Earth from outer space. The video is very experimental, so the thoughts that people report are very broad, so it's not directing your thinking in any direction, but it's rather trying to expand your thinking and in what usually we refer to as a stream of consciousness. So it's really about this flow of thoughts that I'm interested in capturing and sending into space. The signals that were sent by the SETI scientists in the 70s were all about our rational understanding of life. So if an alien civilization would receive those signals, would imagine that humankind is a very technologically advanced, very scientifically advanced species with a very, very rational mindset. Of course, this is only a very small part of what our mind is about. So I think these messages really misrepresent completely what we are about as humankind. And instead, I wanted to really convey what is essential about our experience of life, which is this continuous shift of thoughts. I was very fascinated by the possibility of communicating something which is really contradictory about our nature, and not being ashamed about it, but rather acknowledge that and try to ask perhaps other beings, are you also like that? Do you have the same experience of life as we do? In this project, the concept of dualism is multiple, in the sense that it's all based on dualistic meanings. So the dualistic meaning of body and mind, the dualistic meaning of outside planets and planet Earth, but also about the many contradictions that are part of our lives. The project is based a lot on the mirroring effect as well, which is looking at outside in order to look inside. So it's about exploring outer space in order to understand ourselves better.
Hello, everybody. Thank you so much for joining us for Tektite 2020, Women of Sea and Space. My name is Dr. Tierney Thys. What a day we have had. From meeting with the Aquanauts of Tektite 1 and 2, including the original all-female team of Mission 6, hearing their first-hand accounts and learning from about the upcoming film Tektite Revisited, to learning how the cutting-edge space technologies from NASA and the German Space Agency and COMEX are helping us understand not only the cosmos, but our own inner space, our ocean. Together, we've taken a sonic expedition onto the Great Barrier Reef to listen to climate change, and entered the 3D mucus homes of midwater animals called larvaceans. We've learned about EKGs for coral reefs and coral animals, and about the individual personalities of tool-using tusk fish. Fish are so much more than just protein. And our minds have been opened by thinking about ocean memory and ocean sentience and liquid semi-living systems that iteratively respond and evolve, kombucha-derived clothing for space and sea missions, and ephemeral hyperobjects that shape our perceptions. We have contemplated what is the definition of life and the meaning of it. And it's only day one. So I hope you've enjoyed today's event. And I invite you all to join us again tomorrow for another full day, starting bright and early at 9 o'clock Eastern Standard Time. We'll see you there. Thanks so much.